Wonderful. Good afternoon, and I want to welcome you to the UC Davis Center for Neuroscience's NeuroFest event, which is our annual Brain Awareness Week event. I feel a little funny not wearing a mask up here, but we got special permission from the university for the speakers. We've all taken antigen rapid COVID tests, and so we're all negative today, and so they gave us a dispensation to not have a mask on. I'll introduce myself. My name is Kim McAllister. I'm the director of the Center for Neuroscience, and I'm really happy to see all of you here today in person. The turnout is incredible. It's been a couple of years. We saw some of you online for our online event last year, and of course 2 years ago we had to cancel our event, our NeuroFest event. So as you all know, it's been really a challenging couple of years. And so I just want to take a moment to acknowledge all of the people that are still suffering from COVID around the world. And our hearts really go out to the people of Ukraine who are suffering unspeakable atrocities in the Russian war. So given all of the things that are going on in the world, I am especially grateful that you're all here and that we get to share some of the advances that we're making in neuroscience here at UC Davis. So as we get started, I'd like to extend a warm welcome to a few special guests in the room. So we have Distinguished Professor and Dean of the College of Biological Sciences, Dean Mark Winey, right here. We have Distinguished Professor and Dean Emeritus and major supporter of neuroscience for many, many years, Ken Burtis here. And we have Distinguished Professor and Director of the Center for Mind and Brain and one of the masterminds in starting the Center for Neuroscience, Dr. Ron Mangan. And then we have a number of members of our College Campaign Leadership Council that are shown here. We have Deb Neff, Ron Smith, Steve Constant, and Lynn Weaver there with Dean Winey. So thank you so much for coming today. Okay, so this is really an exciting time at the Center for Neuroscience because we're marking our 30th anniversary this year. And the Center for Neuroscience was established in 1992, 30 years ago. I'm going to give you a little history about how neuroscience has grown up at UC Davis because we have an incredible strength. We have a lot of centers. But it started with some visionary faculty that were from the med school departments of neurology and psychiatry, and a lot of faculty in the Department of Psychology and the then zoology department, because this was before NPB, or neurobiology, physiology, and behavior was established. And these faculty got together and they decided that they wanted to start a neurobiology group. And at most universities about 30 years ago, departments of neurobiology, neuroscience were just starting to be established across the country. But these scientists here decided that they would rather do a different kind of organization for neuroscience. Rather than have just one department, they wanted to have an interdisciplinary center where they could bring together faculty from many colleges and schools who could work together across scales to address the general mechanisms of how the brain works. And so that was the vision for the Center for Neuroscience. We're actually located, a physical center, we're located in Research Park here in Davis, and we've got 3 buildings, and it's completely full now and really a vibrant place. And I would welcome those of you that haven't been to the center to let us know, and we'll give you tours there. But neuroscience isn't just at the center, so things started there. But about the same time that the center was established 30 years ago, the Alzheimer's Disease Research Center was established here by receiving a very large grant from the National Institute of Aging. They have grown, and they're one of the most successful centers at the university. The Mind Institute was, within a couple of years of the center being established, was then built at the School of Medicine. And the Mind Institute, of course, is world-renowned for its investigations of the mechanisms underlying neurodevelopmental disorders. And then we had the Center for Mind and Brain, which really allowed UC Davis to expand from how the brain works into how the mind arises from the brain. And so they provided a bridge, an important bridge to social sciences. Then we had faculty that started to be recruited to a number of different departments because neuroscience has only grown. And so the director of the California National Primate Center was brought in, and neuroscience has been expanded there. We have a lot of neuroscience faculty in vet school departments, in many different med school departments and campus departments, so that now we have a real strength in neuroscience of over 300 faculty, and it seems to be growing every year. So this is very exciting. And we feel that the Center for Neuroscience really provides this excellence in basic neuroscience research that provides a strong foundation for a lot of the really amazing translational work that's taking basic, it's too loud. I can't control the volume. Okay, I can try to talk a little bit lower. I'm sorry if I'm yelling at you guys. We can't control the volume on these things. And that one's not on. So maybe I'll step back from there.
Slow down. You're a little bit fast.
Oh.
Sorry.
Okay.
I know you're excited.
Okay. So the excellence in basic neuroscience research, we think, is really providing a foundation for the translational work that's being done in all of these other units. But because of this distributed nature of neuroscience research across campus, it's important to have programs that brings the neuroscience community together, hence NeuroFest. But we have more formal programs as well, and the center has really been leading in that because it's part of our charter to bring the neuroscience community together. And so we're really excited to have made a very large accomplishment in this regard this past year, where the leaders from all of the different neuroscience centers and departments came together to form this UC Davis Neuroscience Consortium. So the consortium unites more than these 300 diverse neuroscience researchers at UC Davis from 12 centers, 41 departments across 5 colleges and schools to really leverage the strength, breadth, and depth of one of the largest neuroscience communities in the world. And so if you have not checked out the website, the URL is right here. It is a really, really neat website. If you have an interest in a particular topic, you can search for that topic. If you have an interest in particular diseases, you can search for that disease, and you can find all of the faculty that are working within that particular area. We also have a lot of information on the site about the extensive training programs that are going on across campus at UC Davis, from undergrads all the way through every level of training. And you can find a lot of the information about the programs that we have in enhancing diversity, equity, and inclusion, and outreach, which are extensive. Of course, you're here at NeuroFest for one, but there are many other examples. Okay, so at the Center for Neuroscience, though, as I mentioned, it's a physical center. We have 24 faculty that are housed there. It is interdisciplinary, so we have these faculty come from 8 different departments across 4 colleges and schools. There are almost 200 graduate students, postdocs, and lab staff at the Center for Neuroscience, and there are 93 affiliate faculty that are part of our center and participate in a lot of our programs. We also house a neuroscience graduate program, which is growing every year. We now have 60 neuroscience graduate students, and a lot of them you saw out at the booths, that they're really terrific, really excited about the research that they're doing. So as the interdisciplinary hub for neuroscience research and education at UC Davis, the center really brings together neuroscientists from across multiple disciplines, schools and colleges, to advance discoveries. Every day, our neuroscientists conduct cutting-edge research at all levels of the nervous system, from genes to behavior and throughout all stages of life. So we're committed to revealing the remarkable complexity of the brain and leveraging these discoveries to promote health, to catalyze new treatments and technologies to improve lives. So in honor of our 30th anniversary, we have a really exciting program for you. Today's presentations will highlight 4 major areas of neuroscience. We're going to focus on neurodevelopmental disorders, mental health, memory and Alzheimer's disease, and recovery of function after injury. And these are 4 of the strengths, I would say, here at UC Davis in general. So you're going to get to hear from 4 3 at least, great faculty members who are going to tell you about the research that is going on in their labs. Now, typically the talks only focus on the research that's going on in their labs, but this year we have a little bit of a different structure. In honor of the 30th anniversary, we've asked our speakers to talk about where the field was 30 years ago and how the field has changed over the past 30 years, to talk about how their research is where that is focused on right now currently, and then to talk about what the outlook might be for the next 30 years. So you're in for a treat because you're going to get an overview of all of these different areas. So for those of us that perform research, and I see a number of students and postdocs in the room here, and a lot of faculty, it really feels like on a daily basis that we're going at a snail's pace. We want to make discoveries so much faster than we can. We know the need for our discoveries, and none of us, I think, ever feel like we're actually doing enough fast enough. But it's been really fun, and I hope the other speakers feel this way too. It's been fun to look back over the last 30 years and see that it's not a snail's pace. In fact, the rate of discovery has only been accelerating. It's really pretty remarkable how many accomplishments have really been made in each of these fields over the last 30 years. And that really provides a lot of hope for the future. As the pace of discovery is accelerating, I think you'll see that for a lot of these different fields and a lot of these very serious diseases, we're really on the cusp of making discoveries that will lead to new treatments and really will finally improve lives for those of us and many of our family members that suffer from brain disease. Okay, so throughout the afternoon, you can go over to the neuro activities. So I hope you saw the booths as you came in. I don't think you could miss them. They're in the main hall, but we also have darkroom activities and kids room activities. And these are available throughout the entire event. And we have really an incredible team of over 40 graduate students that are volunteering their time. They're so excited to tell you about what it is that they're doing, to share what they know about the brain, and it's really, for me, it's the highlight of this NeuroFest event. So we also have brought back NeuroBlitz, which was the highlight of our online event last year. And you're going to have an opportunity to hear talks from 3 graduate students as the program progresses, and you'll hear more about what the structure of that NeuroBlitz competition is in a few minutes. And then we also have a new neuro art competition. So I saw many of you know that you have a ballot in the materials you were given with the brochure, and we have all of our neuro art entries. There are 8 of them out in the hall. Don't forget the ones that are down at the end. And we're looking for a competition to find our favorite 3 of those. And you're going to hear more about that competition again as the program moves on. So it really takes a team of dedicated faculty, staff, students, and volunteers to make this event a success. And so before we even get started, I want to acknowledge the people that have put in so much work to do this. I have the NeuroFest co-chairs shown here, John Gray and Kostas Sarbalis, who John is over in there, and Kostas, can you wave? There we go. So you're going to see them a little bit later, and they're going to tell you about NeuroBlitz and NeuroArt. We also have our graduate student leads, Becca Wilson and Porter Harast, who I think are out in the booths, making sure that everything is working well. So if you see them, they have been absolutely instrumental in making sure that all of the booths are working well. Then we also have our staff, Teresa Gear, Kimberley Cummings, Katrina Spridgen, and of course Jennifer Scott, who many of you know. Without all of these staff members, we would not be able to make our event work at all. So for those of you that are in here, thank you so much. And if you see them, they're wonderful people. They're staff at the Center for Neuroscience. So say hello. I also want to take this opportunity to thank our donors and CNS Director Circle members whose generosity really allows us to do our work effectively and allows us to move in more innovative directions than we would be able to in our labs otherwise. And so if you're interested in joining our family of donors, we have information at the information booth for more information about the Director Circle, and you can contact Jennifer Scott. Jennifer, why don't you wave? Great. Okay, so now we're going to start our program. In a minute, I'm going to introduce our first two speakers, but I want you to know that after each talk, we're going to have time for questions. So if you've got questions, write them down and raise your hand at the end. And Jennifer and Kimberley Cummings, Kimberley, wave your hand. They're going to come by and bring a mic. We need for you to wait to get that mic because we're actually recording the whole session for people that couldn't make it. And so we'd like for your questions also to be recorded there. Okay. All right, so it is my pleasure to introduce our first speaker. Dr. David Segal is a professor of biochemistry and molecular medicine within the UC Genome Center and the UC Davis Mind Institute. Dr. Segal's research focuses on genetic and epigenetic therapy for the treatment of neurological disorders. And his lab examines how nature causes lifelong changes in gene expression to develop, and he develops epigenomic editing tools that can be used for the long-term control of gene expression for both research and therapeutic applications. Using a variety of delivery systems, he's working to de-risk genetic therapies for Angelman syndrome and other related disorders. And in addition to his research program, Dr. Segal is also the co-chair of the Integrative Genetics and Genomics Graduate Program, and he is truly a devoted mentor. I know, because I have one of his students in my T32 training program. So when Dr. Segal is not conducting really cool research or mentoring his students, he lives life adventurously. So be sure to ask him about his 9-month cross-country walk from Savannah, Georgia to San Diego, California. It's pretty something. So before Dr. Segal comes up, I'm going to go ahead and introduce our second speaker as well. Dr. Christina Kim is Assistant Professor of Neurology. She's going to tell us about how the use of laser light can be used to control brain activity and control behavior, and how this innovative approach may have profound implications for regulating behavior in mental health conditions. Dr. Kim is one of the center's newest faculty members, so she just joined us last September. And her research focuses on developing molecular and optical approaches to study function and organization of individual neurons within the brain. So Dr. Kim's approach is really innovative. She uses synthetic biology, 2-photon microscopy, and animal behavior to deconstruct the building blocks of neural circuits that are essential for motivation and for behavior. So in her spare time, Dr. Kim enjoys long rides in her motorcycle, and she can often be found climbing rock faces in Tahoe. So now, join me in welcoming our first speaker, Dr. David Segal.
Well, good afternoon, everyone. I hope you can hear me. It's a real pleasure for me to be able to speak to you today at this NeuroFest and tell you about the work that we're doing here at UC Davis. And so to get started in thinking about neuroscience, let's talk about cancer. So cancer is a big problem. Cancer affects a lot of people, and probably many people in this room would know someone that was touched by cancer. And so cancer is a big problem, and it deserves a very big response, right? So in the 70s, an American president actually declared war on cancer, which channeled a lot of money to cancer research and the development of cancer therapies. Years later, another American president would launch the moonshot, which is another big boost to cancer funding to raise awareness and to make new therapies and continue on the fight against cancer. And if we look now at the National Institutes of Health, and there'll be a test on this later, so please pay attention. What we see is the largest institutes at the National Institutes of Health that funds a lot of biomedical research across the United States is the National Cancer Institute. It's actually the largest one because, you know, of all the research being done there. And the second largest institute is actually the National Institute of AIDS and Infectious Diseases. And we all know that infectious diseases are a problem because we're all wearing masks these days, right? So clearly, I mean, these are big areas that deserve a lot of attention. Now, suppose I were to tell you that rare diseases, and by rare diseases, I mean diseases that affect less than 200,000 people in the United States. Suppose I were to tell you that rare diseases impact more people than cancer and AIDS combined. So I'm going to tell you about a rare disease that we work on today, but there's about 7,000 of these so-called rare diseases. And in contrast to what I was telling you about before, where the level of interaction is on the level of the President of the United States, people suffering from these rare diseases often have actually very little. Because there's 7,000 of them and they're not individually large, it's hard to get funding to work on these. It's hard for drug manufacturers to actually be interested enough because they're not going to make a lot of profit treating such a small group of patients. It's hard to find investigators that are going to build their careers about working on a disease that maybe affects 25,000 people in the United States. And so for people that are suffering from these diseases, they don't have a president advocating for them. Oftentimes the diseases don't even have a name, they just have the name of the gene. And many of them actually have not a lot of hope. Because these gene disorders tend to have big effects on development, about 50% of people affected by these rare diseases are children. And most importantly, about 95% of these rare diseases have no kind of approved DNA therapy. So this is a major, major unmet medical need. I've been associated with the Angelman community now for a number of years. Angelman syndrome is probably something that many people in the audience have never heard of. And that's good, because it's a terrible disease. So people that have Angelman syndrome suffer a lot from cognitive deficits. On a neurologic level, we think that basically there's a profound learning problem that the problem in this disease prevents these children from learning. One of the things that they never really learn to do is speak. So a lot of them will have very difficult to develop language. You can see that this student or this individual is using a touch screen, which is the way that many of them will be communicating with their families. There's characteristic movement disorders. Some people will be ambulatory and be able to walk. Some will only walk when they're 13. Some of them will be wheelchair bound throughout their lives. Seizures is a major problem. And some Angelman, we call them angels. Some angels will have no problem with seizures. Some will have multiple seizures per day. Some of them will be controlled with seizure medication. Some will not. And they're happy. Actually, happy is part of the symptoms of this disorder. And in the community, they say that, this is one of the things that really keeps the community going, is the happy mood of these children. So what is it that's actually causing this problem? So when the disease was first characterized in the 60s, of course, they really didn't know. They could look at children. They could see there was a syndrome. They called it Angelman syndrome because they didn't know what the gene was. They didn't really know what the causes were. But with advances in genetics, which have been substantial over the last several years, it's now getting to be a lot easier to look through the entire 3 billion base pairs of the human DNA and find those genes that are mutated in those children. So we now know what the mutation is that causes Angelman syndrome. And actually, that's been known for over a decade. But since the problem is on the level of the DNA, we need a solution that's a therapy that can act on this level of the DNA. And until recently, that's not been easy to do. So you probably know that the DNA in our bodies have genes that code for a piece of RNA, and that RNA goes on to make a protein, and the protein is the thing that actually does a job in the cell. And so for people with Angelman syndrome, they're missing an important protein because there's a mutation and this whole process doesn't work. Now, on the level of proteins is where most drugs have their effect. So most drugs are actually drugging a protein. And I'm really happy that we have drugs, but drugs, unless we're fighting an infectious disease, drugs often don't really cure people of a disease. They need lifelong treatment with this. And drugs really can't change a genetic mutation, right, which is the root cause of this. So we would really like to get at this genetic level. And so I'm going to talk to you about advances that have led to some ways now that we can actually start to work on this level of the DNA where the problems are. And what I hope to show you through this is that by developing these tools and methods that can be used to address problems on the level of DNA, we can start to make a program that can work for many of these rare diseases. So making changes in the DNA of living organisms actually goes far back. Some of the early techniques, we were able to manipulate genomes. So an example might be where we use some kind of a chemical that can intentionally mutate DNA in a very kind of light way. And then, maybe we could treat the seeds of a plant with some kind of a mutagen and then grow up the plants. And you might get some big plants and some little plants. And then you can go back and try to figure out what those genes were that made the plant big or made the plant little. And that's how we learned a lot about what genes did. And we still use these kinds of methods today. But that was kind of the state of the art up until kind of in the early 80s when scientists, including Mario Capecchi here, in looking at how nature manipulates its own DNA in our cells, realized that he could use this process that has a long name called homologous recombination. And by studying how that process worked, he was able to make a system whereby he could introduce a so-called donor DNA and set up the experiment so that he can get, for example, this green box to insert itself into a very precise place in a genome, in the DNA of a living organism. And this was a real advance, okay? This gave us really for the first time a way to specifically target a specific gene or a specific part of a gene. So this went on to create things like knockout mice and allowed us really to allow mice to become the preeminent model for human genetics. And we learned all about what our genes do by using techniques such as this. However, these techniques didn't work very well outside of mice. And even with all the things that they were able to accomplish, it still only really works correctly in about 1 in a million cells. So this is about where I walked into this picture. I was a graduate student, and I went to the University of Utah to work with Dr. Capecchi here. And I called him up before I got out there, and I said, can I come and join your lab? This is really interesting stuff. And he said, sure, if you're really interested, any professor would take you in their lab, I'm sure. So I went out there. This is before he actually won the Nobel Prize for this methodology. And I said, well, I'm here now. I'd really like to work in your lab. And he said, actually, I'm sorry, we already took someone from your class. You'll have to work somewhere else. I was like, oh, wow. But I ended up working for this guy, Dr. Dana Carroll. And he was interested in this process. And what he really wanted to know is, why does this only work in 1 in a million cells? And so during my Ph.D. work, a long time ago, what we found out was that the slow step in this process was making a double-strand break in the DNA. That is, to break this DNA molecule. And we found out that if you could break that DNA molecule in a very particular place, you could stimulate this, this insertion of the green box in exactly that right place by hundreds of thousands of fold. So this was what set off really the early days of what we now call genome editing. And it set up this intention to try to develop better nucleases that are more targetable and this kind of thing. So that's going to be the cargo, if you will, that we want to deliver to try to fix these genes that are in people's bodies that are mutated. And to make this a therapy, we need not only that cargo, which is going to be our gene editing system, but we also need a delivery system that can get it into the cells. And so as I'm going to tell you about some of the things that we're working on, keep in mind that these are the two big areas, right? We have these new tools that we can work with the DNA, but first we have to get those tools to the DNA in the body. And when we're talking about neurologic disease, that's in the brain. So as far as the delivery system, we're able to borrow from the gene therapy field. And by gene therapy, what I mean is gene replacement therapy. So in gene replacement therapy, the idea is if you have a cell that has some kind of a mutated gene in it, you could use a virus, because we all know that viruses are good at infecting people, right? So we could use some kind of a viral vector, get rid of all the viral genes, pack it with the gene that we want, a healthy copy of this gene, and now using this viral vector, we can deliver a healthy gene into that cell. So that's what we might call traditional gene therapy. And I'm very happy to stand before you and tell you now that gene therapy is now a thing. And the first gene therapy to actually be approved in the UK was in 2012, and the first gene therapy to be approved by the US FDA happened in 2017. So not many years ago. For most of my career, this was always like an aspirational, someday, hopefully, I think in some time in the future, we're going to have these therapies. But today I can stand in front of you and say, we have gene therapies. We don't have a lot yet, but now there's a path, right? There's a path that we know how to follow, and there are these FDA-approved gene therapies for a small number of diseases. So this is no longer a would-be, should-be, oh, I hope it can happen kind of thing. This is a now thing. Gene editing, what I've been describing to you, I think is becoming a thing. We don't have any approved gene editing therapies yet, but there are some that are in clinical trial. And so the tools for doing this now have evolved over the last 30 years, over the last 20 years, and most of it happened just in the last few years. In fact, of these different types of targetable nucleases now, perhaps some people have heard of CRISPR. CRISPR has been in the news a lot. Even people in my family know CRISPR. They don't want to know a lot about what I do, but they know about CRISPR. Jennifer Doudna and Emmanuelle Charpentier just got the Nobel Prize for CRISPR. So CRISPR is one of these nucleases, and we use this a lot. And it's able to make these double-strand breaks in DNA. And we can design experiments that will either allow that process to make a new mutation in the DNA, so we knock out a gene's function, or we can use it to actually go in and correct a mutation in a cell. So that's going to be our cargo. The first ex vivo gene editing clinical trial actually started in 2009. So ex vivo means that the way that we do this therapy is we could remove cells from a patient, do this editing treatment in a culture dish, and then reintroduce those cells back into the patient. So this first clinical trial was actually done for HIV. Now, the HIV virus has to bind to a couple of receptors on the surface of the cell, and it turns out that one of those receptors is not so important for function, for day-to-day function in people. And so they were able to show that they could knock out that receptor so that that receptor is no longer on the surface of the cell, and the HIV virus essentially just bounces off the surface. So like this, they've been able to actually treat people that have HIV, correct this, make their cells HIV resistant, and now they can have a functional immune system and not develop AIDS. So this is still in clinical trial. the first use of this CRISPR nuclease in a person, so now this is not in a culture dish, this is in a person, just happened in 2020. So this stuff is like just happening right now. This is in clinical trial. The first one was actually treating a rare eye disease where they injected into the eyes. And we can go back and talk about why they were injecting into the eyes afterwards. But now you can see the stakes are a little higher, right? Because now you're not doing something in a dish where you can control the conditions so much. You're injecting this into a person, and that editing is going to happen in a person. And you want things to go right, because anything that doesn't go right, it's going to be hard to go in and fix it again. So, quite appropriately, this technology is moving a little bit slower. But these are in clinical trial. And when I say clinical trial, that means they're testing it in people. Now, there are some concerns that people have raised about, whether this kind of gene editing, making these breaks in the DNA and having them reseal and getting the right information in there, how well all that's going to go, and are you going to introduce new mutations in this process? So we don't want to introduce any new mutations, right? We already said that mutations are the problem. So some people are trying to think about what is the next thing. So I've told you about gene therapy that's now been approved, some of them approved by FDA. Talked about gene editing, now in clinical trial, testing it in people. We hope some of them get approved through FDA. Our lab is working on what we think is going to be the next thing, which is this epigenetic editing. Now, don't be afraid of this word epigenetics. All I want you to know about it is that, if this black line kind of represents this long DNA molecule, epigenetics is little chemical modifications to those DNAs, where particular chemicals inside our cells attach themselves to the DNA. So it's not changing DNA sequence, it's just making little chemical attachments to our DNA, and that sends signals to the rest of the cells to tell a gene to be turned on or to be turned off. This is part of our natural way of controlling genes in our cells. But, in doing many studies about this, we know the enzymes that are responsible for writing those kinds of chemical marks, and we know the enzymes involved in erasing those kinds of chemical marks. So now we can use something like CRISPR to now direct those epigenetic information to be deposited on a particular gene. So basically, we gain the ability to tell a gene to be turned on or to tell a gene to be turned off in a very precise way. And we could introduce that with this viral vector technology that gene therapy is using. So we're really honing in on getting to a very safe, not going to make mutation kind of way to control this gene expression. And we're applying that for disorders like Angelman syndrome. So this is a picture of many people in my group. We have an angel here. This is Jason. He was visiting our lab here at UC Davis. And we've had many angels visit our facilities. And, for many people in my lab, this is not anymore an academic exercise. We like to be part of this community. We like to get involved. We like to see the people and find out what they care about, what they're going through. We were very fortunate. A lot of our funding has come through a foundation called the Foundation for Angelman Syndrome Therapeutics. They took a very aggressive approach to funding. So it turns out that actor Colin Farrell here has a son with Angelman syndrome. And he became the kind of celebrity face of this organization. At this gala event that they have every year, he brings his entourage. This is Jai Courtney, this is Greta, some other movie stars he brings with him. And they were able to raise lots of money for research and to build awareness. When I joined up with them a long time ago, even many pediatricians never heard of Angelman syndrome. Today, they have three drugs in clinical trial. So this is a tremendous, tremendous advance in the history of this disease. I can't claim to have been responsible for lots of that, but Angelman is way ahead of most genetic diseases, right? I mean, they have a lot of research going on. There's 27 companies now that have Angelman syndrome therapeutics in their pipeline. But again, for many diseases, it's nothing like this, right? But we're working as part of this group. And so we work very closely with 2 other labs here at UC Davis. One is Kyle Fink, that's shown in this picture, and another one is Jill Silverman. And so our 3 labs work closely together. My lab is mostly making these kind of therapy parts, the epigenetic editing parts. Kyle's lab is trying to test them in human cells so we can make the human therapeutic, and Jill Silverman's lab is testing them in mice. And so we need all of these parts to really get this work done. So here's an example of just something that we're doing for this rare neurologic disease of Angelman syndrome. We can build one of these epigenetic editors. It does its thing on the DNA. We can inject it into a mouse. Then we can do some experiments and see that the gene is now turned on. That's this green color after treatment. And we could see it in another kind of assay. That's on the molecular level. Then we can look with electrophysiology and ask if the neurons are working better. And this data tells us that they are. And we can even do some behaviors. I told you that these Angelman kids have movement disorders. They have trouble walking. The mice models of this disease also have trouble walking. And working with Dr. Jill Silverman at the Mind Institute, we were able to show that the mice are able to walk better after treatment. So we're actually seeing improvements to the symptoms from this kind of treatment. So this is for a rare neurologic disease. We even have some aspirations that this can be applied to even more common neuropsychiatric diseases such as schizophrenia, autism, depression. Now those are more complex diseases, involve a lot of genes, involve a lot of interactions with the environment, so a more complicated place to work in, but affect a lot of people as well. And all I would say with this is that we know that some early life stresses can, for example, in the mother, could predispose their children to end up with some of these kinds of conditions. And we know that epigenetics has something to do with that. All the dots are not completely connected on how that's happening, but since we can make these epigenetic tools that work in the brain, it's kind of, thinking way into the future, maybe not so far into the future, can we actually, to the extent that early life stresses encode some epigenetic changes in the DNA of these kids, can we actually go in and reverse that information so that now their condition will be more like a neurotypical condition than constrained by these epigenetic changes. So we have a lot to learn on that, and we have some funding to work on this too. But we think that we might be able to impact these areas as well. So I could just kind of stop with this provocative statement: can we erase a bad experience using molecular biology? Well, we'd like to see if we can. So I do feel, in closing, that there's a strong need for an academic center for interventional genetics. A lot of work has to go into these kinds of studies and developing these treatments. I don't know if the Wall Street way of making medicine is going to work here, where investors want to see billion-dollar returns on their investments to treat these disorders, because they're not like cancer. They don't affect hundreds of millions of people. They're affecting smaller groups of people. So how are we going to solve this unmet medical need? So we're trying to do that. Our labs are down in this kind of section of these little bubbles on translational disease research and therapy makers. But it really takes a village of people all collaborating together: clinicians, regulatory people, industry partners, philanthropic support, bioethicists. It takes all of this to really make this more than just working on our own, but coming together and mustering our resources to make therapies that could really impact the lives of people. These are some of the people in our village that are doing this work. So this was actually on a plane. We were going to a conference, and so it has some people from Kyle Fink's lab, some people from my lab, and these are some people in Jill Silverman's lab. And some of our sponsors and places where this is done in different labs on the Davis campus and the Sacramento campus. And so we hope that this work will lead to outcomes not only for Angelman syndrome, but for many other neurologic diseases as well. And I thank you for your attention. Question there.
Thank you for your wonderful talk and very clear explanations. I had a question. There was a moment when you talked about the different angles of the research that you and your two colleagues were doing. One looks at cells, one looks at mice, and your lab looks at the other perspective. I didn't catch what it was, but I was hoping that in the future this would be a framework from when I'm reading about a certain topic, and I would ask myself who is doing the research from those three angles.
Yeah, so you're asking who's doing that research?
No, what was the aspect of your research
Oh,
that
the
contributed to those other two angles?
Right, so our lab focuses on the kind of underlying technology for how we would do it. So, you know, for the case of Angelman syndrome, we're trying to develop something that can actually silence a particular RNA transcript. So we would try to design that part on a molecular level and demonstrate that you know, the required erasers and writers of this epigenetic information are functioning properly. Then we would hand one off to the person that's working on the human one and another one off to the one that's working on the mouse one. We often have to develop these in parallel because the DNA sequences in mouse and human are sometimes different. So we have to take these two parallel tracks so that we can show that it can improve the symptoms in a mouse, but we end up with a human targeted therapeutic in the end as well. So we really have to work in close-knit combination.
Thank you.
Question here.
Hello, thank you so much for your talk today. I just wanted to ask really quick, what was the specific mutation that causes the Angelman syndrome?
Oh, great question. So the mutations occur in a gene called UBE3A. And we know that from what similar proteins do in that class of proteins, we think it has something to do with degrading proteins. It's interesting to note that that information about which gene was mutated and what that gene does didn't help us at all to come up with a useful therapy. So, you know, we always like to think, like, if we could only find the gene responsible, we could find a cure. Well, we knew the gene. It didn't really help because the connection between what those children experience and what we know that the gene does is very disconnected. So, you know, other people are trying to research, you know, why is the loss of that gene causing all these symptoms like lack of speech? But today we don't know that. But what we do know is that there's this mutation, and so we start out with a very simple hypothesis that if we could replace that gene function, things should get better.
We have time for one more question.
I have a question about if you, in a disease that's passed on down from parent to child, if you repair the gene in the parent before the parent has a child, will that fix the problem of something like sickle cell anemia going from one generation to the next?
So yes, it would. If we could fix that gene in the germline cells, that's like sperm and egg, of the parents, then they wouldn't pass it down to their children. However, this falls into this realm of germline gene therapy or gene editing. And because that type of therapy is not treating the person that has the disease, but actually ends up changing the DNA of someone that would be born, at this time, that kind of therapy is considered to be too risky because the changes that you would make then would be inherited by this person. And until the science underlying it becomes more mature, there's a general consensus that we should confine this technology to people that are sick and not to people that are not sick, but we hope to avoid disease in the future.
So when you treat the person that's sick, do the physical symptoms go away?
That's going to depend on the particular disease. So in Angelman syndrome, the answer we believe will be yes, for various reasons that I would be happy to go into separately at some point. But that might not be true for all diseases, and this is an ethical consideration that you bring up. Yep.
Thank you, Dr. Segal.
Thank you again.
Thank you all so much for being here today. I'm honored to be speaking here. This is my very first NeuroFest, and I'm looking forward to attending as a community member in the years to come. And so today I'm going to be talking about something that's probably been on a lot of our minds over the past couple of years, which is our mental health and how we can think about improving our mental health. And so mental health conditions, whether it's a very serious mental condition you suffer from or whether you're experiencing symptoms of one, can really affect our daily lives, right? So we can experience symptoms such as anxiety, feelings of hopelessness, feeling antisocial, et cetera. And this can influence the way that we sort of experience the world, make it hard to engage with family or friends, or even make it harder for us to do our work. And these symptoms, while some of them can, rather than just one of these symptoms being assigned to one specific type of mental disorder, they're actually shared among many different types of conditions that we think and talk about. And a key issue that our neuroscience research is trying to address is how can we treat these symptoms and conditions to improve our daily lives and the lives of patients that are suffering from these conditions. So first, I just want to give you a very brief history of kind of what the first types of antidepressant medications were developed. And one of the first ones really came about in the 1950s from sort of an unexpected side effect of a completely different type of drug. And so this is a drug that in the 1950s was being prescribed to treat TB or tuberculosis. So this is an antibiotic. And what patients were reporting is that many of them that were under this medication were experiencing side effects of euphoria. And patients that were suffering from depression were actually reporting that they could feel their symptoms kind of going away after being on this medication. Around the same time, there was a completely different drug that was being used to treat blood pressure. And this had the complete opposite effect. Patients under Reserpine were reporting that, hey, actually, I'm starting to feel withdrawn, and many of them were saying, I'm starting to feeling symptoms of something like depression. And this led people to wonder, okay, what are these drugs actually doing in the brain? They're not supposed to be doing anything, towards our mental health. These are supposed to be treating physiological disorders. What's going on? It turns out that what these drugs were doing is altering levels of different types of neurochemicals in the synapses between our brain regions. And so this TB medication was actually increasing the levels of these neurochemicals in our synapses, and that was associated with euphoria, whereas this blood pressure medication was actually decreasing the levels of these neurochemicals in our synapses, which was causing patients to feel perhaps symptoms of depression. And from this sort of serendipitous finding, this TB medication was actually one of the first antidepressants that were being prescribed to try and treat this disorder. So studies like these led to this early theory, this sort of prevailing theory that many people have held for a long time, that many mental health conditions or neuropsychiatric diseases are caused by something like a chemical imbalance in the brain. So you may be familiar with that term. And some of these neurochemicals that are thought to be involved include serotonin, which you may sometimes hear people talking about, oh, I need a serotonin boost or some energy boost to help me get through my day. Another neurochemical called dopamine. This is thought of as the pleasure molecule. And also another one called norepinephrine, which is important for alertness or maybe focus. And the primary one of these neurochemicals that are targeted by the most common antidepressants that we use today is serotonin. And so you or a loved one may be familiar with these types of drugs. So for example, Prozac and Zoloft. And these are pharmaceutical drugs that end up increasing the levels of serotonin in your synapses. And so I think these drugs have been tremendous at treating depression in many, many patients. However, in about 50% of patients, they report that drugs like these have no effect. So this is perhaps called something like treatment-resistant depression. And in fact, sometime in 2008, there was this big review study saying that actually drugs like this, antidepressants, which we thought were going to be a panacea, kind of a cure-all for many mental health conditions, weren't as effective as people thought they were. And so this led neuroscientists to start to look at other theories beyond this just neurochemical imbalance theory of what's going on in the brain underlying mental health conditions. And another theory that people like to think about is, okay, rather than just focusing on the neurochemicals, what if we think about what are the actual neurons doing? Like, how are they firing in the brain? And maybe in people suffering from mental health conditions, they have abnormal neural activity patterns in certain brain regions. And so then this inspired this line of research, I think, asking, can we just reset these abnormal patterns of neural activity that we see in these patients to try and help these symptoms? And so in 2008, we received FDA approval for an approach called transcranial magnetic stimulation. And this is a way that we can try and stimulate neurons in one particular region, the dorsolateral prefrontal cortex. And this, in patients that are normally completely unresponsive to pharmaceutical drugs, has a massive improvement in their symptoms when they're suffering from treatment-resistant depression. Another type of approach for this stimulation is deep brain stimulation, or DBS. This was first FDA approved for Parkinson's in 1997, and since has been approved for dystonia, epilepsy, and obsessive compulsive disorder. However, it's not yet approved yet for treating major depression disorder. And this is because there are still ongoing clinical trials trying to pinpoint the exact neuroanatomical location that you need to implant these electrodes in to have the best sort of efficacious effect in humans. And so I'll pause here and I'll just recap some of the treatments that I've described that we developed over the past 30 years. So on the one hand, we have these pharmaceutical drugs. And these drugs target a very specific chemical, for example, serotonin. However, they'll target that chemical everywhere in the brain. On the other hand, we have these brain activity stimulation approaches such as DBS or TMS. And these are great because they can focally target one particular brain area, like the dorsolateral prefrontal cortex. However, we're not quite sure what the stimulation is doing. We don't know exactly which neurons are getting increased or inhibited, and it's targeting every neuron in that brain region, regardless of the type of neurochemicals it receives. And so I think right now we're at a place where we're in a dire need for new types of therapies that can treat these debilitating conditions. And ideally, they would have high efficacy, they would be fast acting, and they would be non-invasive so that they could help as many patients as possible. And so how can neuroscience research in the lab, in animals, help us create better therapies for something like depression or other mental health conditions? So a very first critical question is, what are the specific neurons that we need to hit and that we need to target in order to eliminate these symptoms of these mental health conditions? But not only that, if we then want to go and target them in as many people as possible, it would be great if we could understand what their biochemical makeup is at the protein level. You just heard about that in the previous talk, so that we could develop drugs that can target those very specific neurons that are involved. And the reason that we can do this, approach these questions in the mouse, is because in animal models, we have very precise ways and new technologies that allow us to stimulate a precise subset of neurons that we think are relevant for causing a particular set of symptoms. And one such example of a technology that was developed in 2007 in my graduate advisor's lab is called optogenetics. And so before I tell you about how optogenetics works, I'm just going to give you a very quick primer on how the neurons in our brain naturally communicate and fire. And so when two neurons are speaking together, one neuron will first send a signal and cause the release of these neurochemicals that we've been talking about. These neurochemicals will then bind to ion channels that are expressed on another neuron. And then these ion channels get activated by these neurochemicals. And that can trigger an influx of sodium into your cells, and this is what causes neuronal firing. Ideally, we would want a way to not have to rely on this neurochemical release to trigger that sodium influx and neuronal firing. We want something that could just cause the neurons to fire whenever you want. And this is the approach that allows us to do that, and we call this optogenetics. And it's really been kind of a game-changing approach in neuroscience research that was just developed as soon as 2007. And so the core technology of optogenetics was this discovery of light-gated ion channels, or opsins, and these are naturally found in microbial organisms in nature. And so what we can do is we can express these opsins in neurons so that they just sit at the membrane where other ion channels normally sit. And then we shine them with blue light. It activates these ion channels, and that causes positive ions to flow in. And this can trigger that same exact mechanism of causing the sodium influx and the action potential firing. So it's great because we can kind of just bypass one step and trigger neural activity with light. These opsins exist in all different types of flavors, so you can have inhibitory opsins. When you shine orange light into the brain, it causes negative ions to flow in, and this can block naturally occurring neural activity. So the way we can use this in animal research is using a lot of the same approaches that we just heard about. You can take the DNA that encodes an opsin, package it into a harmless viral vector, and inject that virus into the mouse brain. How do we deliver light into the brain? For now, we can implant a very small and flexible optical fiber into the brain, and then that can be connected to a cable that just delivers blue light or orange light. So I'm going to show you a video now of this in action. This is one of, I think, the very first times that I saw optogenetics working in a behaving animal. And so this is a mouse that's been injected with one of these excitatory opsins in the motor cortex. And when the researchers shine blue light into the brain, you'll see it light up in a moment, the animal immediately starts moving. And so this is happening in real time and is a result of directly stimulating neurons in the motor cortex. And you'll see as soon as the light turns off, the mouse will stop moving. So this was pretty remarkable to see this happening. And then, of course, we started to wonder, what other brain regions can we then target and stimulate and see how that affects behavior? And I just want to show you another really elegant example where it's something more complex than just simply moving around. But this is doing something like driving some innate drive of feeling thirsty. So here they're targeting a very small subpopulation of neurons. And whenever they stimulate them, the animal immediately runs to its water bottle and starts drinking water, even though before it was completely uninterested in drinking. And when they turn that light off, the animal stops drinking water, stops feeling thirsty, and goes about its normal business. So this is a level of control that we can have now on neural activity and seeing how it affects behavior. So the question is now, can we apply approaches like optogenetics to think about how can we treat symptoms in animal models of mental health conditions like depression? And luckily, many of these behaviors that we see in humans, we can model to some approximation in animals. And I'm going to briefly share 2 examples with you today about anxiety and a feeling of loss of pleasure. So how do we measure anxiety in a mouse? So one example is something called the elevated plus maze. And so this is a plus-shaped maze that we kind of sit high above the ground. And two of the arms of the maze have walls, so that when the animal is in those sides of the arms, it feels safe, it can't see anything around it, it's nice and dark. And then the other two arms are completely open, so there are no walls, and the animal can kind of peer over the edge, and it's a little more anxiety-producing for them. So when you do this assay, if a mouse tends to stay in these enclosed arms, this can correlate with something that we read out as feeling anxiety. And then when an animal's less anxious, it'll feel more exploratory, and it'll go out and start to look at the open arms, peer over the edge, et cetera. In 2015, researchers found that when you stimulate neurons in the prefrontal cortex in a rodent that are expressing this opsin, they'll become less anxious. So they'll actually, in this very same task, rather than spending most of their time in the enclosed arms, suddenly they'll start going out and exploring these open arms and being much more curious and active. A year later, another study was trying to look at how can we measure something that we call anhedonia in rodents. So anhedonia is just a fancy word for when people feel a loss of pleasure. So normally things that made you feel happy, that brought you joy, you no longer want to do them anymore. The way we study anhedonia in rodents is through something called a sucrose preference test. So there are two water bottles in its cage. It can either drink the plain water or it can go after the sugary water that they really enjoy. And people had found that in genetic animal models of depression, the animals will exhibit a much less lower preference for this sucrose. So this is kind of a readout of anhedonia. And what this study found is that when they optogenetically stimulated the prefrontal cortex, very precisely to when those numbers in blue correspond to the days that it's receiving this optogenetic stimulation, just on those days, the animals would exhibit less of a sucrose preference, indicating that they're experiencing this anhedonia. And so wait a minute, I just described to you two studies, 1 year apart, where in one case they found that optogenetically stimulating PSC could reduce symptoms of something like depression. It makes the animals less anxious. And then another study found that it worsens symptoms. It makes the mice feel anhedonia. So what's going on? And it turns out that these are different populations of neurons in this region. And they're sending signals and projections to completely different brain areas in the mouse brain. So if I wanted to go in and try and target this brain area to improve depression, I would want to increase the activity of this purple pathway, while I would want to decrease or lower down the activity of the pathway in the red. And so without approaches like optogenetics that we're able to do in the rodents, we never would have been able to tease apart this kind of very subtle pathway distinction. And so this is why these types of techniques are so important for looking at the circuitry. And so hopefully from what I just described to you, I've convinced you that neuroscience research in animals can help us answer this first question. What are the specific neurons that are relevant? Where are they in the brain? Where do they project to? And these are the ones that we want to target. But then the next big challenge is, okay, let's say I know the neurons, but how am I going to actually influence their neural activity, right? In a human, thus far, we don't have a way to pinpoint and say, okay, I'm going to deliver deep brain stimulation just to the neurons in one region that send axon projections here and not there. The technology is not there yet. So instead, to help translate these findings into humans, perhaps we can target them using drugs. And so this is where I think something that a lot of people are starting to work on over the next, and we'll see improvements in over the next 30 years, is can we develop drugs or antidepressants that can recognize just a subset of neurons that we think are important for driving symptoms of depression. And in order to do that, we need to know at the protein level what are the specific targets in these neurons that we need to develop drugs and biochemical markers for. And so this last part of this puzzle is something that my lab, my new 6-month-old lab at UC Davis is working on. And so the way that we're approaching this problem is by first using bioengineering and tool development to try and think of new technologies that allow us to then target, to figure out what proteins are expressed in functionally defined neurons. And then we employ these technologies along with those like optogenetics and mouse behaviors to test whether they allow us to target relevant neurons. And then we can use genetic approaches such as molecular profiling and something called proteomics to actually learn what the proteins are expressed in these cells. And through collaborations with many different centers and other researchers at UC Davis, we can apply these 3 different approaches to ask, how does the brain regulate mood? Specifically, my lab is very interested in looking at this one pathway that I highlighted before. So this is from the prefrontal cortex to actually another region called the nucleus accumbens. And we think that perhaps in patients experiencing certain types of mental health conditions, this pathway is maybe upregulated or overactive. And it would be great if we could figure out a way to dampen down the neurons just in this pathway and see if it improves symptoms of depression. One of the ways in which we're trying to tackle this question is, again, through technology development. So this is a new approach that we developed, I guess, a couple of years ago now that we called Flicker. And it's a new type of optogenetics that allows us to detect the neurons that are active during some behavior and then tag them with a molecular tag. This tag can let us know, okay, which neurons were actually activated while the animal was experiencing some scary or aversive experience. Then we can isolate just those relevant neurons and then do molecular profiling so that we can ask what proteins are expressed in these cells. And then we're going to ask, can these specific proteins we found serve as drug targets to reduce symptoms of depression, first in an animal model and then moving forward? And so the hope for the next 30 years, right, we just summarized a lot of progress that's been made. But the hope looking forward, I think, is first, I expect there to be still a lot of emphasis on new drug therapies to try and treat different neurochemical pathways that are thought to be involved in depression. So for example, psychedelic research is something that's really being spearheaded by professors here at UC Davis. Ketamine as well is a brand new sort of recent treatment for treating depression as well. But on the other hand, I think that we're going to see a really massive improvements in these types of interventional stimulation technologies to actually alter the brain activity inside of humans. And I don't think it's that far off to think that, in maybe 30 years, we'll actually be at the point where we can do something like non-invasive optogenetics in humans. And so in 2018 was actually the very first clinical trial using optogenetics in the retina of a human to restore sight to a patient that was blind and suffering from a hereditary disease where their retinal cells were dying. And so this is just one example of, once we slowly kind of work out, I think, all the safety and the ethical concerns, it's one way that we may see this technology translate into treating human disease. And so that, I think, really what I wanted to emphasize and what I've hopefully been able to share with you today is the need for interdisciplinary research where rather than just focusing on neural stimulation and neural activity or just focusing on what is this certain neurochemical that we need to target, by combining and sort of bridging these two aspects of research, I think that's going to be critical and crucial for leading us to a better understanding and eventually an actual cure for these types of mental health conditions I've told you about today. And so even though we are brand new here at Davis, I've really been able to get settled and form a sense of community here already. There are 2 of my students in the back taking pictures. But I'm thrilled to be starting my career here. I'm thrilled with the support and the community here. And I'm happy to take questions now from anyone about our research.
Thank you. Hi, thank you for speaking. Oh, here I am. I'm just curious about if you were to induce long-term potentiation using optogenetics, would it create or could it possibly be used as a way to create like long-term behavioral change without the use of optogenetics afterward? Thank you so much.
Yes, so the question was whether, rather than having to use optogenetics all the time to sort of change neural activity and stimulate neural activity, could you use it to drive synaptic plasticity, something long-term potentiation? So maybe you could just drive plasticity once and then see these long-lasting, long-term effects in neural circuitry and brain health. I think that's a great idea. I haven't seen anyone really start to do that yet, but it's something you could precisely test in a mouse and see, months later, years later after we've done the plasticity effects, do we see long-lasting changes? That's great.
I just had a question. What about certain light therapies that are on the market? Would they be similar to what the same effects that you're discussing? Some of them to treat seasonal affective disorder, even just decreasing the blue light on your phone.
Yeah, exactly. So this is a good point. So some people have, there have been studies that have shown that blue light alone, for some reason, I don't think we know the mechanism yet, but blue light alone is able to actually alter your neuronal activity. And so I think more recently there was one where they'd shown that patients treated with a certain frequency of blue light that seemed to improve, I think, their memory in an Alzheimer's model, and a couple of other studies as well that have found that. So that's a great point. I think because we don't understand how that mechanism works yet, or which brain regions it's actually changing neuronal activity in, optogenetics is trying to be a more precise way. Let's say I know for sure I need to hit only the neurons in one brain area and hit them with a certain color of light. If you were just delivering blue light by itself, it might cause nonspecific effects as well.
Hi, Dr. Kim. Thank you for your talk right here. I just wanted to ask if you could touch on the advances that are going on with psychedelics as drug therapies. I know it's like a kind of hot topic and controversial field, but I'm curious because I have seen some stuff in the news, especially about the ketamine research, but not about LSD or psilocybin.
Yeah, so the real expert in that is going to be Dr. David Olson, whose entire lab is really focused on asking how can psychedelics help treat symptoms of depression, and he's also developing and synthesizing new chemicals right in his lab that are trying to sort of pinpoint the same chemicals, the same receptors, but without the hallucinogenic effects, right? So that's a major safety concern of using these psychedelics, maybe the hallucinogenic effects, whether those are necessary for improving symptoms or not, that's a major line of research. And so my lab is we're actually collaborating with Dr. David Olson, and so we're looking at how activity in the prefrontal cortex changes when you inject 5-MeO-DMT or a newly synthesized analog TBG from David Olson's lab. John Gray also is an expert in thinking about how these psychedelics can affect the pharmacology of neurons. And so it's an active area of research, and I think Davis is really sort of leading the way in that avenue.
Last question?
I think we had one here. Yeah.
So you didn't talk about ECT at all. And can you just tell us where that falls between the deep brain? I mean, that obviously has been done for many, 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 many years.
Exactly. So I failed to mention, so I wanted to say before even DBS or TMS were starting to be used in clinics, before that, I think maybe even before the 1950s, electroconvulsive shock therapy was being used to try and reset the neural activity and reset the brain activity. And I think that was really crucial for sort of establishing this theory and testing this theory and seeing it work in certain patients. And then from there, the goal moving forward, I think, has been trying to figure out less invasive ways to get the same results, the same improvement in symptoms, but in a more focal manner and a maybe more non-invasive manner. So the TMS is trying to do the same thing, but without actually, with a magnet to alter neuronal activity rather than delivering these more powerful electrical shocks.
Why do you think it's being used more widely now than it used to be with gerontology patients? What is the difference between using it with a young person and a old person?
I'm not too sure about that. It might be something about just the mechanism in which these two different approaches work, how effective they are at actually changing the neural activity. And so if it's something developmental, one approach works better in one versus the other, that may be a reason why. Yeah. Okay.
Kim.
Okay, thank you.
So thank you so much, Dr. Kim, for an absolutely fantastic talk. That was really stimulating. So now I'd like to introduce Dr. John Gray, Associate Professor of Neurology and co-chair of NeuroFest, to share a little bit more about the Neuro Art Contest that I know a lot of you were enjoying before the program started. So, Dr. Gray.
I want to let you guys know that outside these doors, we have a number of really beautiful images. These come from the various research labs here at the Center for Neuroscience and at the broader UC Davis neuroscience community. And this is part of our Neuro Art Competition for this year. This is something that we started back in 2020 to really get some beautiful images to be in our lobby of the Center for Neuroscience. So this year we brought it back. We had 45 entries. We picked 8 finalists, which are the ones that you see outside. And in your program, you'll have a ballot. The top half is for the Neuro Art Competition. And we'd like you each to rank your top 3 images that you see and drop it in the box that's out there. We have some students that are manning the box if you have questions. But rank the top 3, and then we'll have awards for the winners. So there's a monetary award, $300, $200, $100 for the top 3 contestants. Many of these were students that have submitted their own images that they've taken in their labs. And also we're going to basically print these in a higher format and use them in our lobby of the Center for Neuroscience. And so it'll be a permanent part of our gallery. So let's take some time and look at those images. In addition, if you're interested in seeing our gallery or just visiting the Center for Neuroscience, we have a place where we can, out there, where you can sign up for like a small group tour. Somebody can show you all these images that we have in these various competitions and show you some labs at the Center for Neuroscience. So consider doing that. So now we're going to take a 30-minute break. You guys can go back and look at the booth some more. You can take vote on your favorite images. There's some beverages and snacks outside. When you're outside, you can only eat those outside. You can take your mask off when you're out there, but please put it back on when you come back inside. And then we will come back here at 3:30 for the NeuroBlitz, which will be some excellent student short talks and a few more faculty talks. So feel free to ask questions and enjoy the rest of your day.
Okay, everyone. Welcome back from the break. I hope that you had a chance to go and see more of these booths. I see a lot of the yellow shirts from our graduate students. It's so much fun out there, and I've heard a lot of great feedback. So I thanked all the students when you guys weren't in here. Let's give them. Oh gosh, I forgot about that. I'm so used to having a mask on. This is why it's great that the university allowed us to do this by having the rapid test. So anyway, what I was garbling under my mask was that I've heard a lot of great feedback about all of the graduate students that are running the booths. You guys are doing a phenomenal job. I thanked you when you weren't here, but let's give them a round of applause. Okay, so we are thrilled to bring NeuroBlitz back by popular demand. We started NeuroBlitz last year during our online NeuroFest. And so now I'd like to invite Dr. Kostas Zarbalis up to the podium to share more about what the NeuroBlitz competition is. First, let me tell you a little bit about Dr. Zarbalis. He's the co-chair of NeuroFest, and he's an associate professor in the Department of Pathology and Laboratory Medicine. He's also the president of the local chapter of the Society for Neuroscience, and we're deeply grateful for the chapter's sponsorship of this portion of our program. So, Dr. Zarbalis.
Everybody can hear me? Yeah, apparently. Well, thank you, Dr. McAllister. So this is my second year that I'm working with the students for this NeuroBlitz presentations, and I'm very proud of this year's contestants, as I was also last year's. So NeuroBlitz challenges our graduate students to sum up their research in a concise and succinct talk, similar to an elevator pitch that is geared towards a general audience. So each student will have 3 minutes to present their research. Our student assistant, Kaylee, and I'm not sure who Kaylee is. Is it you? Hi, Kaylee. Will keep time and let our speakers know when their time is coming to a close. So as a member of the audience, your help is requested too. We need you to vote for your favorite NeuroBlitz presentation and on how well the students, and basically assess how well the students did in engaging the audience and how clearly they communicated the significance and concepts of their research. Once all of our contestants have been given their brief talks, please take out the ballot in your program. You should find one there, and cast your vote for your favorite NeuroBlitz. So the student with the most votes will win a $200 award, which is sponsored by the Society for Neuroscience's local chapter. The winner will be announced by the end of the day. And now let me introduce this year's NeuroBlitz graduate student contestants. So the first contestant is Jessie Badley, who joined the UC Davis Pharmacology and Toxicology graduate group in 2020. She studies the neurotoxic effects of developmental exposures to lower chlorinated PVCs using primary neuronal cell cultures. Jessie currently trains in Dr. Pamela Lyne's laboratory. So the second contestant will be, is unfortunately Jasmine Cardin, and only unfortunately because she cannot present. So Jasmine had an unexpected family commitment and will not be able to participate. So feel free to cross her name off from the ballot. The third contestant will be Lucas Cuya, also from Dr. Pamela Lyne's lab. So he's interested in understanding molecular and cellular mechanisms underlying neurodegeneration, with the goal of helping patients afflicted with these neurodegenerative diseases. And he joined the Neuroscience Ph.D. program in 2021. And our last contestant will be Stephanie Lozano from Alex Nord's labs, who began her PhD program here in 2020. Her research interests are rooted in understanding the molecular and cellular interactions of the neuroimmune interface in health and disease. So NeuroBlitz is a fast-paced competition, and there will be no time for questions at the end of it. So however, we hope their short pitches will stimulate your curiosity and encourage you to connect with them throughout the day. So I'm sure they will be available for questions later on. And with that, I'm handing the stage over to Jessie.
Hello, everybody. How are you today? So I'm going to be talking a little bit about my research as it pertains to neurodevelopmental disorders. So autism prevalence has increased 178% since the year 2000. And as prevalence increases, so do the costs associated with medical and non-medical services like education, early behavioral interventions, even caretaker costs. In a 2020 survey done by the CDC, they found that 1 in 54 people was diagnosed with autism. But when we dig deeper into the why, research has shown that no single genetic anomaly is the answer. It's actually a combination of gene and environment interactions. We're truly products of our environment. And even though we might not be able to point the finger at one specific gene, we can do things to mitigate these risk factors in our environment. So let's talk a little bit about brain cell development. There's this incredibly important window of time when brain cells called neurons are working hard through a series of developmental stages. And research has shown that maternal exposure during this time actually can impair maternal exposure to environmental toxins can actually impair fetal brain development. And so depending on when this exposure happens, this can lead to an imbalance of brain connections. The input and the output don't match, essentially. And this is a common trait in many neurodevelopmental disorders, including autism spectrum disorder. So one of those environmental toxins that I'm very interested in is a group of synthetic chemicals called PCBs, polychlorinated biphenyls. Legacy PCBs were used heavily in production of building materials that we see up here, like paints, caulkings, building sidings, pesticides, newspaper inks, and they were used very heavily up until their ban in 1979 for having cancer-causing properties. And although they're no longer in production, we're finding new contemporary PCBs popping up, and they're still very much a problem. They're being found in our food, they're being found in water, they're also being found in our schools. These PCBs also showed up in a UC Davis marble study in the serum of pregnant mothers that were at risk for having another child with a developmental disorder. So our lab has begun testing these compounds and found that PCB11, one of the major contemporary PCBs found in that marble study, actually increased the number of input points. So there's going to be an imbalance of connectivity. But PCB11 isn't the only contemporary PCB that we found. So insert me. This is where my research starts in the lab. And my focuses are essentially on the who and the how of developmental neurotoxicity of other prevalent contemporary PCBs. So by understanding these mechanisms of these chemicals and how they affect our bodies, especially the developing brain, we can identify future interventions, ways to mitigate exposure, and also potentially shape public policy so we can get some of these risk factors out of our environment. So I just want to give a quick thank you to my lab and my lab mates for all of their support, the NIEHS for my T32 funding, and also my graduate group, PTX. Thank you.
Hello? Okay, cool. Neurodegenerative diseases can be devastating. They're often associated with increased age and affect over 6 million people in America alone. This number is expected to rapidly increase due to our continuously aging population. Despite their prevalence, meaningful therapeutics remain few and far between. Clearly, more research needs to be done to better understand these diseases so that we can develop ways to help people affected by them. Neurodegenerative diseases can be characterized by neuron death, accumulation of pathological protein, and neuroinflammation, or activation of a brain's immune system. This is the piece that we'll be focusing on today. So the immune system in neurodegenerative diseases, is it good? Is it bad? Well, similar to my love life, it's complicated. Let's get into it. The science, that is. The immune response, you know it, you love it. It's there to protect your body from foreign invaders, harmful pathogens, virus, and bacteria. But you may also know that this protection comes at a cost. Inflammation, fever, pain, fatigue. For the most part, these symptoms are natural side effects of the immune system doing its job and protecting you. Perhaps a more apt metaphor might be, instead of a shield, a defensive flamethrower. But generally speaking, the good outweighs the bad, especially when this immune response is short-lived. Perhaps you're laid out for a few days, but then you get better and the virus is no more. Let's talk about the brain. This is your brain. When you were young, a blank slate. But as you got older, you started developing cellular debris, even if you're otherwise completely normal and healthy. It's just a byproduct of living. The cellular processes in your brain generate waste, which are largely degraded or otherwise recycled. But as you get older, these processes become less and less efficient, and you start generating more and more protein junk. At a certain point, the immune system will be called into action to try to clear this potentially harmful protein aggregation, causing inflammation. While it does clear it, there are those negative side effects I mentioned before. But you keep living, so you keep generating more debris, and we keep trying to clear it with our immune response, and this goes on in a state that we call chronic neuroinflammation. When we have this persistent activation of the immune response in the brain, that balance between good and bad, protection and harm, becomes a little bit less clear. And if you have certain genetic risk factors, or perhaps have a previous history of brain injury, then the harm may outweigh the good and could potentially cause neurodegeneration. My research is interested in understanding the mechanisms behind this balance and trying to shift it more towards protection than harm, so that those at risk for these diseases and have them can live happy and healthy lives.
Okay, hello? Okay. All right, so autism spectrum disorder is a neurodevelopmental disorder that affects 1 in 44 children in the United States. People with autism exhibit a number of different physical, behavioral, and cognitive differences relative to their neurotypical counterparts, including but not limited to the ones shown here. As the name suggests, those with autism fall on a spectrum. People on the severe end of the spectrum may be so impacted by their autism that they are unable to live their lives independently. Useful therapeutics have been developed to help these individuals, but due to the complex nature of the disorder, what works for one person very well may not work for another. Now, the symptoms shown here are differences we might expect to see on the outside, but they can all be traced back to differences on the inside, differences that start at the level of cells and DNA. My research focuses on what's happening at the cellular level. We want to know, how do differences in an autistic person's DNA or RNA collectively give rise to the symptoms we see at the larger level? Now, autism genetics are incredibly complex. It's not like a mutation in a single gene will cause you to develop the disorder. Plus, there's also environmental risk factors to consider. However, the severe form of autism that my lab is interested in is very strongly driven by genetic makeup. So much so that if we take a mouse embryo and mutate a single autism risk gene called CHD8, the mouse will go on to develop symptoms that are similar to those seen in humans with severe autism. Before I get into how we use those mice to study severe autism at the cellular level, let's go over the 3 main types of molecules that help our cells do all that they do to keep us alive. Within our cells, we have DNA, which is transcribed into mRNA, which in turn is used as instructions to build proteins, the workhorses of our cells. I like to think of this in terms of baking a cake. The DNA is the recipe that you found online. The RNA is like the recipe that you printed out, maybe made some changes to. And the proteins are like the cake itself. Now, without going any deeper into that analogy and leaving you all hung up on the thought of chocolate cake, there's another key idea I have to mention here. And that is that genes within our DNA can be turned on or off. This is what DNA looks like inside the nuclei of our cells. If a gene is turned on, it will be transcribed into mRNA, which will go on to help make a protein. If a gene is turned off, no mRNA is produced and thus no protein. This means that RNA is a readout of gene expression. So if we sequence an individual's RNA, we can tell what genes were turned on at the time the RNA was collected. What's more is that different cells will be expressing different genes, and therefore will have different RNA or gene expression profiles. We can see that demonstrated here. Each of these colors represents a different gene that's expressed at a different level in each cell type. This is the sort of output we'd expect to see from a technique called single-cell RNA sequencing, which is different from standard RNA sequencing in that it allows us to see which RNA is being expressed by which cell type, instead of an average of RNA expression across all cell types. Getting this single-cell resolution was not possible until relatively recently. It's super important to have single-cell resolution because with genetically linked disorders like autism, some cell types may be affected while others are not. And this will be evident in the RNA-seq data. With data like this, we may be able to identify which cell types are being disrupted to ultimately give rise to large-scale autism symptoms. Then later, if we want to develop genetic therapies to help with autism symptoms, we can try to make it so that the therapy only targets the cell type that needs it and not the others. Now, how does my lab in particular use single-cell RNA sequencing to study the cellular basis of autism? I mentioned before that we have a mouse model of severe autism that has a strong genetic influence, and we generate these mice by mutating an autism risk gene called CHD8. What we can do is collect RNA from the brains of wild-type mice, or mice with no mutation, as well as mice with a mutation, and then go through cell type by cell type, comparing the RNA profiles to see if there's a certain cell where we detect a disease-relevant difference. This is exactly what I've begun to do. So far, I've generated single-cell RNA-seq data, And using these data, we're able to identify a bunch of different cell types. This is a representation of cells in dimensional space. Each dot represents a distinct cell that we collected RNA from, and this is for a mixture of wild type and CHD8 mutant cells. If the cells cluster together in dimensional space, we can say they're the same cell type. What I plan to do next with these data is look for differences in RNA expression of wild type versus mutant cells, and see which specific cell types may be altered in the mutants. If we can determine what cellular processes are disrupted in the mutant mice, we may have a better idea of which cells are involved in autism pathology. And this may ultimately help us develop therapies that target those specific cell types while leaving the others alone. That was a lot of info in a short time, but thank you for listening, and thank my lab and the rest of the neuroscience grad group.
That was exciting, right? So please join me giving one more time a round of applause to all the contestants. And for the next let's say 8 to 10 minutes, we'll vote for, please vote for your favorite NeuroBlitz using the ballot in your program. Once complete, please pass the ballot to the person to your right, and our team will come by to collect them from the person at the end of each row. So while our team finishes collecting your ballots, please allow me to introduce the next speaker. This is, at a young age, she has always been fascinated with science. She started out as an amateur botanist and ornithologist, until she read a book about memory research as a freshman in college and never turned back. Today, she's the first female director of the Center for Neuroscience and a professor of neurology and of neurobiology, physiology, and behavior. She's the co-champion of the UC Davis Healthy Brain Aging Initiative and a principal investigator of the UC Davis Conte Center, funded by the National Institute of Mental Health and one of only 15 such centers in the country. Her research examines how circuits in the developing and aging brain are formed and sculpted. Her lab is one of the few in the world that studies the role of immune molecules in regulating synapse formation and function, as well as the molecular pathways that underlie risk and the resilience of offspring with brain disorders following maternal immune activation. She's a trailblazer not only in her own research, but also through her mentorship and training of young researchers, advocating for diversity, equity, and inclusion in science. Please join me for in welcoming Dr. Kimberley McAllister.
Okay. Can you hear me better? That's better, okay. If it is too loud or difficult to hear, could somebody in the back raise their hand and we'll figure that out? There's all sorts of feedback that we're getting from a bunch of these different sound systems, so I really apologize for that. Okay, so I am going to start off my talk with a very simple question. At least a simple sounding question. What is memory? As I get older, when I think about this question, I actually think more about what is forgetting. And I think about all the things that I forget all the time, increasingly as I get older, right? Where's my iPhone? How many times have you said that? I say it all the time. Where's that darn TV remote? Where did I put that? I don't remember. And the biggest one for me now, where did I park my car when I come out of a store, right? So as annoying and as common as these examples are, they really don't illustrate the real importance of memory. The ability to learn and remember is fundamental to every single act of daily living. Our memory enables us to learn from the past, to stay oriented in the present, and to plan for the future. So as Stephen King put it, a person's memory is everything, really. Memory is identity. It's you. So who am I? I am a daughter, I'm a sister, I'm a mother, and as you've heard, I'm the director of the Center for Neuroscience. Without my memories, I would not be me. I would be me, but I wouldn't have my identity. And that's honestly the harsh reality of what people who suffer from Alzheimer's disease and dementias face. So this picture, which is similar to the picture that Lucas used in that beautiful talk a few minutes ago, really poignantly illustrates the loss of identity that accompanies Alzheimer's disease. And as Lucas pointed out, this terrible disease is way too common, and it's way too devastating. Alzheimer's disease is the 6th leading cause of death in our country, and it's the only top 10 cause of death that cannot currently be prevented, treated, or cured. As Lucas mentioned, it's affecting about 5 million people right now in our country, but this rate is only going to increase. It's going to increase, they estimate, to 7 million by 2030, and a whopping nearly 14 million by 2050. So we clearly need to develop new treatments as fast as we possibly can to stem the suffering that we know is coming in a tsunami of disease by 2050. So how can we develop approaches to improve memory and to prevent diseases like Alzheimer's disease? Well, there are a number of simple steps that we must take in order to achieve this goal. And a lot of it involves looking into the brain to identify where memory actually occurs in the brain. Second, we really want to determine then how memories are established within circuits within those brain regions. Third, we want to identify how memory loss actually occurs in disease. And then, fourth, most important, we want to develop strategies to treat and prevent memory loss in disease. So the strategies that we think we will be able to identify in step 4 really rely on the other steps. They rely on allowing us to come up with ways to fix the things that go wrong in disease that we'll identify in step 3, using approaches that are identified in step 2 to restore the typical way that memories are made in the brain and within the very specific brain regions where memory actually occurs. So where does memory occur in the brain? About 30 years ago, we did know a little bit about where memory occurs. And when the center was launched 30 years ago, our basic knowledge about where it occurs came from historical studies from psychologists who had studied patients that had lesions in the brain. And they had lesions based on either injury or on surgical resection for treating diseases like epilepsy. And some of the most important discoveries were made by an inspiring scientist, a psychologist named Dr. Brenda Milner, who's shown here on the slide. And she studied one particular patient who had a really fascinating type of memory loss. Some of you may have heard of this patient. He went by his initials, we refer to him by HM to protect his privacy. Now this man had intractable epilepsy, and to treat that epilepsy at the time, what they did was to resect a region of the brain that I'm circling here on the slide, right around what we now know as the hippocampus. And so that area was completely missing in HM. The surgery improved his epilepsy, but it led to a major problem with memory that was really interesting. HM was no longer, after surgery, able to make memories for new events. But he could remember events from his past that he had had before the surgery. And so what Dr. Milner concluded from this was that memories for new events must occur in the region of brain that was taken away in the hippocampus, but memories for past events must somehow not involve the hippocampus anymore and instead be stored in this area of the brain outside of the hippocampus. Okay, so since HM, our field has developed really great techniques that allow us to look into the brain in healthy individuals to see where memory occurs. And so functional magnetic resonance imaging, or fMRI, was developed actually only about 30 years ago. We all just assume it's been around forever, but it was only 30 years ago that people started using this. And it's one of the most widely used approaches to start to study where memory occurs in the brain. Now we have several faculty members here, some in the audience, who use this approach and who are leading world experts in human memory. And at the Center for Neuroscience, one of our experts is pictured here, Dr. Charan Ranganath. So fMRI has begun to reveal the activity patterns that are involved in creating and recalling a specific memory. As memory is encoded and initially retrieved, the hippocampus lights up, which is this area that I'm pointing at right here with the pointer. But long-term memory storage occurs across many different brain regions depending on the memory. Different types of memory involve different sets of areas. So for example, long-term memories for words might be stored in the front of the brain, whereas long-term memories for scenes might be stored in the back of the brain here. Now, Dr. Ranganath's lab, he asked me for a favor, and they actually need some help with soliciting volunteers. So his lab is interested in understanding where these different types of memory are encoded, but he really wants to understand how that encoding and that long-term storage changes over healthy brain aging. And so we know a lot about what happens at the end of life, we know a lot about what happens early in life, but we don't know much about what happens over the process of healthy aging. And so he has a study that he needs to recruit volunteers for. So if you are over age 60 and you're interested in being part of a neuroscience study, please let me know. We've got flyers down here, and Dr. Ranganath's lab would be really happy to have you volunteer if you're interested. Okay, so once we start to have a sense for where memory might be encoded, we want to ask the question of where does memory actually occur within those brain regions. Now, the search for the physical manifestation of a memory in the brain, which is called the engram, has lasted for more than 100 years since the engram, that term, was first defined. And it was initially proposed that memories are stored in subsets of cells within specific brain regions that are connected to each other. And so in order to be able to find those cells, we have to be able to resolve them. But fMRI doesn't have the resolution where we can actually see single cells. And so while fMRI told us where to look, generally within the brain, we had to turn to animal models to start to figure out how memories were actually encoded within those regions. And so this search for the engram has been driven by technological developments, similar to a lot of the technological developments that you've all heard about over the course of the day today. So in terms of this particular search, it involved mouse research. And before 1980, it involved an approach that was really similar to what psychologists had used in studying HM. Basically, scientists removed large parts of the brain and looked to see whether memory was still there or whether it was gone. And over time, those techniques were refined in the 70s and 80s, where they could create targeted lesions of more specific brain regions, narrowing where the memory might be stored. And then in the 1990s, scientists developed ways to silence neurons within a specific brain region, again, now getting closer to identifying the neurons that are actually involved in creating that memory. But it wasn't until 2009 that techniques were developed that really allowed us to start to identify the actual neurons that are part of an engram, of the physical manifestation of a memory. And one of the key advances that allowed us to do that was the development of methods in mice that allowed us to identify the neurons that were active at the time of learning, and then to express genes within just those individual neurons. Okay, so what you're looking at here is a picture, a slice through the hippocampus. This is packed full of cells. You don't see most of them, but you see a subset of these little green neurons. They're really beautiful, right? These are neurons that are expressing a green fluorescent protein that was turned on only in the neurons that were active at the time of learning a task in a mouse. So what's great about this technique is that it doesn't just allow us to turn on markers like green fluorescent protein, but it also allows us to turn on genes that we can use to control the activity of the cells, just like Dr. Kim described in her talk, a beautiful talk earlier. And we have a faculty member that's actually in the room who is very famous, Dr. Brian Wilchin, Brian Wave, and I'm embarrassing him, but he's a member of the Center for Neuroscience, and Brian was instrumental in developing and using this technology to start to identify the engram. And what Dr. Wilchin and his colleagues can now do is pretty phenomenal given where we were in studying memory 30 years ago. What they can do now is actually to go in and not only identify the neurons that are involved in a specific memory, but they can erase memories by silencing those specific engram neurons, and they can induce artificial memories by stimulating those engram neurons. So whether we're going to be able to do this in humans is an open question. And as Dr. Kim mentioned, this is something that people are starting to think about how to adapt these approaches to be able to be used in humans. But the idea eventually would be that in early Alzheimer's disease, as individuals start to lose their memories, we might be able to use these approaches to reawaken memories in those patients. But regardless of whether this will turn into a treatment, it has told us a lot about how memory, where memory occurs, and it's starting to give us some insight into how. So the way that the engram works is that it's made up of connections between a small subset of cells within a number of different brain regions. These are initially hooked up to the hippocampus, and as memories are made, they fire together, they are active at the same time, and that strengthens those connections. Over time during recall, as the memory is recalled over and over again, that then leads to strengthening of the connections in the areas of the brain that are important for storage, and the removal of the connections to the hippocampus. And that really begs a really interesting question. How can the strength of some connections be modified during memory formation and storage? Other connections from cells that are neighboring not be affected at all, and how can it lead to the removal of other connections? So in the 1950s, one of the most influential ideas for how this might work was proposed, again by another psychologist, Donald Hebb. And Donald Hebb proposed that neurons that fire together wire together. And so what that means is that cells that are active during the formation of a memory will have their connections strengthened so that they remain connected to facilitate the recall of that memory. Now, it wasn't until about 30 years ago that we started to gain any understanding of how synaptic strengthening and weakening might actually occur. And the process that underlies the long-term modification of the strength of synapses is called synaptic plasticity. And there are lots of forms of synaptic plasticity, but two in particular, long-term potentiation strengthens those connections and long-term depression weakens them. And so the synapse should be familiar to you now after the talks earlier. Basically, the way this works is that information is transferred across cells by the release of neurotransmitter that then binds to receptors on the recipient cell and therefore transmits information. When these connections are stimulated rapidly, like would be happening in a memory, you end up with an increase in synaptic strength or potentiation of your connection, which means that basically it's releasing more neurotransmitter and there are more receptors to bind to, so the signal is just a lot louder. So it's really actually astounding how much progress has occurred in our understanding of the cellular and molecular mechanisms that mediate this. This is just the tip of the iceberg here. In fact, there have been 17,000 papers that have been published since 1992 just on the mechanisms of long-term potentiation alone. It's a lot of papers. It's a lot of work. So several faculty at UC Davis have contributed to this knowledge, including myself and Dr. John Gray, who studies some of the most important neurotransmitter receptors that are involved in LTP called the NMDA receptors. So one of the most exciting advancements in the field has been the discovery that strengthening connections by LTP during memory formation can also cause more connections to form, which further solidifies that engram. So what you see here is a slice through the hippocampus. You see a number of these green cells. That's just a subset of the cells that are now labeled with this green fluorescent protein. And if you come and look at just this little tiny box here, At higher magnification, you can see that process of the cell, the dendrite, actually has a lot of little protrusions on it. These are called dendritic spines, and these spines are where synapses form onto these neurons. So about 30 years ago, again, scientists developed really sophisticated approaches to allow us to image these structures in living tissue. And another one of the center's faculty, Dr. Karen Zito, developed a way to look to see how these spines change with long-term potentiation. And what she found is that when you stimulate a spine, you can actually, if you stimulate it with a protocol that would be found like during memories or forms, long-term potentiation, you end up with the spine getting much larger, and it stays larger over a long period of time. So ultimately, though, to restore memories, we're going to want to increase the strength of synapses. And to do that, we need to know, as Dr. Kim really beautifully pointed out, we need to know what the components of those synapses are and how those components are modified by synaptic plasticity. So while we generally knew how synapses were potentiated about 30 years ago, we didn't have any idea about how complex these synapses actually were. And we've gained a lot of really surprising understanding of that over the last 30 years. So don't get scared, this is a picture, I'm not going to go through it, but this is a picture of just the tip of the iceberg as far as the complexity of molecules at the synapse. It turns out that there are over 1,000 different kinds of proteins that are found at synaptic connections in, at least just for excitatory synapses in the brain, and there are many different kinds of connections. And so while this looks a little bit overwhelming, and even for those of us in the field, this is pretty overwhelming, the sheer complexity of this is incredibly beautiful to those of us that want to come up with mechanisms to improve memory. And that's because each one of these molecules actually has the potential, if we understand it, to be able to be used as a target for developing new drugs against that can then modify synaptic plasticity. Okay, so how can this information be used to develop new treatments to improve memory and prevent diseases like Alzheimer's disease? Well, of course, first, we need to understand what's wrong with the memory machinery in this disease, and then we need to fix it, right? So for Alzheimer's disease, it's been known for more than 30 years that there is severe degeneration of the brain and loss of synapses in the brains of these patients. But it wasn't until about 40 years ago that some of the causal pathology was actually identified, what actually leads to this loss of brain tissue that you can find in severe Alzheimer's disease. So some of you know about this. You've heard about the plaques and tangles. So it was found in the 1980s that these are comprised of amyloid and tau protein. And it wasn't until about 30 years ago, kind of remarkably, that the first genetic risk factors were identified. I know this because I was at Duke University starting my graduate career when they identified some of the ApoE genes. Also around this time was the very first time that the different stages of changes in the brain during Alzheimer's disease was identified. It didn't even happen until about 30 years ago. And it wasn't until 25 years ago that the first mouse model of a risk factor for Alzheimer's disease was actually established. So this field of understanding what's going wrong in disease has not been around for that long, but it's picking up pace. And so only recently, in the last few years, have we really started to gain insight into what might be going wrong at these synapses so that we can start to figure out how to fix it. And so this is a rather complicated figure that I'm going to show, I'll take you through. It illustrates the results from a study that I find really exciting. So this is a neuronal cell body. This is a normal synapse, so this is where you make connections and you have neurotransmitter release. In the Alzheimer's brain, in an early brain, what you see is you do see the accumulation of amyloid and amyloid plaques. And for many years, it was thought that it was these plaques that were actually causing the degeneration and the disease. But it turns out that it's actually in early disease, it's these little A beta monomers that get secreted from the neurons and that actually go in and they come into the synapse, they bind to molecules at the synapse, and they cause those synapses to be hyperexcitable. Hyperexcitability might sound good sometimes if you want some energy, but it's not good for neurons because neurons die when they're hyperexcitable. And so this is just one glimpse of a new discovery just over the last 3 years of how some of the basic pathology in Alzheimer's might be altering synapses to cause synapse loss early on. But as Lucas told us in the data blitz, it's not just amyloid that leads to Alzheimer's disease. It's also inflammation in the brain. And so about 20 years ago, it was recognized that inflammation plays a role in Alzheimer's disease. But it wasn't until about 10 years ago, only 10 years ago, that the cell type, the immune cell type that plays this role was identified. And these are called microglia. But we know very little about how inflammation causes synapse loss and leads to loss of memories in Alzheimer's disease. So the lack of understanding of how synapses are lost in Alzheimer's has led to a lack of progress in developing new treatments. So current treatments that many of you may be familiar with for mild to moderate Alzheimer's disease are basically anticholinergics like Aricept. To moderate severe Alzheimer's disease are drugs like memantine. But that's kind of it. How can that be? There are a number of current clinical trials that are now focused on developing ways to alleviate the pathology from amyloid. So people are using antibodies to A-beta. There's a recently FDA-approved drug for doing just that is quite controversial in the field. But I think we could all agree that new treatments are definitely needed. New targets are needed. And so a number of us are really interested in synaptic proteins that mediate plasticity and memory and that are altered by A-beta and inflammation, as looking at those as new targets for developing new drugs to treat Alzheimer's disease. And so I've got one more data slide and then I'm going to wrap up. And this data slide basically tells you about why, one of the reasons that I care about this from a scientific perspective, because our lab has just recently moved into this field. And so we are interested not in the immune cells that are involved in synapse loss, but we're interested in immune molecules that are found at synapses and how they can lead to synapse loss. And the molecules that we focus on are major histocompatibility complex 1 molecules. So these are molecules that some of you may have heard of. If you read about immunology, they're found on all of the cells in your body where they help you to fight off infection. A number of years ago, my lab showed that MHC molecules are actually found at the synapse where they regulate synaptic plasticity and lead to synapse loss. So a talented postdoc in my lab here named Gabby Sell has recently discovered that MHC molecules are actually upregulated by amyloid beta in Alzheimer's disease models. And that upregulation leads to a decrease in plasticity molecules at the synapse and causes synapse loss and memory deficits. Now this is exciting because she's now proposed an intervention where she can prevent that increase in MHC, which then might rescue these effects. And she has some recent data that's very exciting, suggesting indeed that's exactly what happens. So if we prevent this increase in MHC, we end up with rescuing the plasticity molecules, they come back to normal levels, and we prevent synapse elimination and memory deficits. Now, we're very far away from looking at this as a treatment, but this is really exciting because once we start to find molecules like MHC that might be able to rescue these deficits, we can then start to spend a lot of intense effort trying to figure out if these are good targets for developing new drugs against. And there are many labs, this is just one example that we're excited about, but there are many labs across the world, hundreds, that are identifying these targets and are following them through to see if we can develop new treatments. So it's very exciting. Okay, so I hope that you can see how far we've come in our understanding of each of the different steps that we need to follow to come up with these new treatments over the last 30 years, and that the pace of discovery really is only accelerating. We've gone from knowing very little about where and how memory occurs in the brain, and even what goes wrong in Alzheimer's disease, to being on the cusp of developing a wide range of strategies to treat and prevent memory loss during disease. Those strategies will include things that you've heard about over a number of talks this afternoon, including brain stimulation, altering activity within the specific engrams, maybe in the future, and developing whole new classes of therapeutics. So the outlook for the next 30 years is finally, I can say, with real enthusiasm and scientific rigor, it really is that we finally have real hope for the millions of people that are going to be afflicted with Alzheimer's disease in the next 20 years. Thank you.
Hi, thank you so much for the presentation. So a quick question on the controversial new drug, the Adulam, I believe it's pronounced. So if the main purpose is to try to reduce the amyloid beta plaque proteins, how come so many patients are developing more neurological issues or even dying? What causes that, and why can't that be circumvented?
Right, so these are the questions that you exactly want to ask when you're doing clinical trials, right? You think that you have a candidate, you test it, and then sometimes you get effects that you're not expecting, which is why we do clinical trials. I'm not an expert in that exact drug or privy to all of the data that underlies that, so I can't answer your question specifically. But I can say that when you alter anything in the brain, you're going to have unintended effects, right? Especially if you're not targeting it to the specific cells that are at the root of the memory loss, okay? So what Dr. Kim talked about is a much more targeted strategy where you wouldn't be affecting all of the cells in the brain, really in the body if you take a drug, but instead it would be targeted directly at the cells that might actually be the source of the issue. Right now, and for the next short foreseeable future at least, we're going to have to deal with this and find and go through a lot of different candidates and hopefully find ones that have less side effects and better outcomes. Yeah, but there's a lot of potential and a lot of things that will be coming down the pike in the future. Yeah. Yeah, one thing that I would also say about that is that I was really surprised when I looked back over the last 30 years, because I thought that we would be, I've been frustrated as to why we aren't further along in having new treatments. But when you look at what we knew 30 years ago and what we needed to know to be able to come up with new treatments, this field is moving fast. We need to move faster, we all know that. We feel that urgency, but we're really, really on the cusp of making big discoveries. And for those graduate students in this room right now, you're going to be the ones that do that. Okay. Thank you. Okay, so I wanted to thank you all for sticking with this. We're getting great questions, and this is just a wonderful meeting so far. But it's time for our last speaker, and the exciting topic of restoring movement and communication using brain-computer interfaces. Dr. Sergey Stavisky is an assistant professor of neurological surgery, and he's co-director of the UC Davis Neuroprosthetics Lab. He's a neuroscientist and a neural engineer who works at the intersection of systems, computational neuroscience, neural engineering, and machine learning. His research focuses on understanding how the brain produces movements and applying this knowledge to build brain-computer interfaces to restore lost speech and movement for people with paralysis that, for instance, might have come from spinal cord injury, from stroke, or from ALS. For 2022, Dr. Stavisky made a New Year's resolution to learn how to cross-country ski and to take advantage of his newfound proximity to Tahoe. And recently, he joined the estimated 14% of Americans who acquired a new pet during the pandemic. So meet Feta, and join me in welcoming Dr. Stavisky.
Hi. Thank you very much. I'm thrilled to have recently joined this UC Davis neuroscience community. And I really appreciate this opportunity to tell you about brain-computer interfaces and how we're using this new technology to restore patients' lost movement and communication. I think many of you in the audience may recognize Christopher Reeve. He was an actor famous for his role as the original Superman. Tragically, he was paralyzed after breaking his neck in a horseback riding accident and became an activist for improving the lives of people with disabilities. Like most people with very severe spinal cord injuries, Christopher Reeve remained paralyzed from the neck down for the rest of his life. The late physicist Stephen Hawking provides another poignant example of how conditions like ALS rob individuals not only of their ability to move, but also to speak. And there are a lot of such individuals. About 15,000 in the US with ALS, and hundreds of thousands with tetraplegia, where stroke and spinal cord injury are the leading causes. What's notable about both Christopher Reeve and Stephen Hawking is that despite their immense fame and resources, there was almost nothing medicine could do to help them. And that's the really frightening reality today. Despite all of our medical advances, if the connections between your brain and the muscles are damaged, we still cannot heal that. However, thanks to modern neuroscience and engineering, we're starting to be able to offer at least some means to regain the ability to move and communicate. So this woman, she lives in the Bay Area and has ALS. She can't talk, she can't move, but she's part of the BrainGate clinical trial of a brain-computer interface, or BCI. And in the video I'll play, she's going to try to move her hand as if controlling a computer mouse. She has hundreds of electrodes in her brain, and together with computers and algorithms, these form this BCI, which allows us to figure out the movements she's trying to make based on her neural signals. She uses this BCI to control the computer cursor and type on screen. I'm just going to turn up the volume a bit. So a BCI can also restore the ability to reach and grasp. In this video from my postdoc work, this gentleman who had a spinal cord injury about a decade before he enrolled in our study is attempting to move his arm and hand. He's got about 200 electrodes in his motor cortex. Note that there are 2 of these little boxes on top of his head. And as you can see, we can do a pretty good job of figuring out what he wants to move and when he wants to move. And so let's unpack how this works. The problem we're trying to solve is that the brain signals can no longer reach the muscles. And so our strategy is to basically bypass the injury and go to the source of the movement command, a part of the brain called the motor cortex. Once we have these neural signals, we apply computer algorithms to identify or decode what they mean. And from there, we reconnect these movement commands to an action. For instance, moving a computer cursor, a robot arm, or even synthesizing speech. And in some cases, we'll even complete this loop and electrically write information back into the brain, as I'll show you later. So a key step in this BCI is actually measuring brain activity. Here I'm showing you the type of sensor used in these earlier videos. Each of these so-called Utah electrode arrays consists of 100 tiny wires, and these wires are placed inside the brain where they can listen to the activity of one or a handful of neurons. And although we're only able to measure a tiny fraction of the millions to billions of brain cells involved in controlling movements, this still gives us a great deal of information about what the person wants to do, if we can understand what this brain activity means. So how did we as a field build up enough information to make such BCIs possible? Well, in keeping with today's theme of 30 years, I want to show how far we've come before turning to what's hopefully to come in the future. So by the end of the 80s, we had a rough understanding of how to interpret brain activity in the motor cortex thanks to work in animal models. And so a lot of the foundational studies look something like this. A monkey would be trained to move this handle to one of 8 targets. And while he was doing this, scientists would use a single electrode at that time to measure the firing activity of one neuron. So here's an example of the data. Each of these little tick marks is when this neuron fired. And these data are grouped by which target along that circle the animal is reaching towards. And so what researchers observed was that the neuron would fire a lot more for certain directions, in this case leftward reaches, and a lot less for reaches in the opposite direction. And so we'd say that this neuron has a leftward preferred direction. So 30 years ago, scientists realized that by looking at the activity of a bunch of different neurons, each with their own preferred directions, we could basically add up how strongly each of them was voting in favor of its preferred direction, and thus figure out collectively which way the monkey was about to reach. And this knowledge formed the basis of the first decoder. So imagine now we have a person with paralysis and we're trying to build a BCI. Let's say we've put an electrode array in their brain and we can measure this example neuron. We would first have the robot arm make automated movements left and right, and then ask the person to try to make the same movement. And I'll now play this animation and you'll hear the activity of a neuron. So that neuron became very active when the participant was trying to move their arm to the right. And this training process lets us calibrate our decoder. So now, if we close the loop and have that robot arm controlled by the BCI user's brain activity, whenever this neuron fires more, it would nudge the arm to move more to the right. And so this approach was finally put into action 18 years ago with the first human trial using multi-electrode arrays. So here we see a man named Matt Nagel, who's the very first participant, controlling a computer cursor.
Excellent. So what's next?
Next, I'd like to draw a circle.
You're going to draw a circle. Wow.
And as you can see, back then the performance was not amazing, right? This cursor kind of wanders around, the circle is not very round. But in the past decade, thanks to more sophisticated decoder algorithms and a better understanding of the nuances of motor cortex, performance improved. So by 2015, it had doubled, and it doubled again in 2017. And so for comparison, on the left, I'm now showing the very fast and precise cursor control demonstrated 5 years ago by the Stanford lab where I did my postdoctoral training. And so if this were used to select letters on a keyboard, this allowed the participants to type at a rate of 8 words per minute. And it gets even better. So just last year, the same group doubled performance yet again, this time by decoding attempted handwriting rather than point-and-click selection of letters. So in this video, you can see letters appearing on the screen as this participant imagines writing them with a pen, even though he can't, of course, move his hand. So I just want to take a minute to emphasize how essential animal research was to making this technology safe and effective for people. The reason I could enable people with paralysis to move robot arms is because we learned so much about how the brain controls arm movements and hand movements from monkeys, which have a similar anatomy. And so here you can see an early study in which a monkey who's not paralyzed is controlling a robot arm to feed himself. Only 10 years ago, these discoveries were translated to people. So I work with clinical trial participants, not monkeys, but there's basically no way that labs like mine would be able to build BCIs for patients if there were no animal studies to prove, first of all, that the devices we're implanting are safe, and also to build this foundation of fundamental neuroscience that informs how we decode brain activity. So what kind of BCI advances do we have to look forward to in the next 30 years? Well, one frontier is to reactivate paralyzed limbs. So instead of using decoded movement commands to move a prosthetic arm, neuroengineers are trying to apply electrical stimulation to move the person's own paralyzed muscles. About 20 years ago, a group showed this was possible. So in this first demonstration, the person's arm muscles were reactivated with wires, but this wasn't done using brain activities. The patient had to press a button on the little external control unit to select different patterns of movement. And about 10 years ago, a group performed another monkey study where they temporarily paralyzed the hand muscles with lidocaine. And they then demonstrated that they could decode the intent to grasp from the brain using Utah arrays and use that to drive the hand muscles. And so finally, 5 years ago, a full muscle stimulation BCI was achieved in a person. This man here is moving his arm with just motor cortex decoding plus wires that stimulate his muscles. Now, as you can see, it's really slow, it's far from able-bodied, but he is feeding himself for the first time in years. And by the way, this contraption over his arm, it's not like a motorized exoskeleton. Rather, it passively holds his arm up because the strength provided by this early muscle stimulator was not sufficient to fully counteract gravity. So in the coming years, we'll see new and better muscle stimulators, and that will allow patients like this gentleman to move much more quickly and to get rid of those wires that go through to the skin, basically make everything fully implanted. A similar approach is also being used to restore walking. So just this year, a group in Switzerland showed that they could implant stimulating electrodes on the outside of the damaged spinal cord. And so when the right commands are entered on this tablet, the stimulator allows this man to stand and to even to take steps. What I think is really amazing about that work is that this group went from a first demonstration in rats 10 years ago to monkeys only 4 years later, and started in people just 5 years ago. And so it seems really likely that in the coming years, if not year, they'll be able to command the walking just using the person's brain activity instead of relying on that external controller. A second BCI frontier is to restore the sense of touch in people who are paralyzed. So the idea is that we can put sensors on a prosthetic limb and transmit information about the limb into the brain by applying precise electrical currents in the corresponding brain areas. So in what should now be a very familiar pattern, foundational work was done a decade ago in monkeys. This read and write BCI showed that animals could tell apart different patterns of sensory cortex stimulation. And 5 years ago, this was translated to clinical trial participants in the same set of groups at University of Pittsburgh. So in this video, when the researcher presses on one of the robot's fingers, the corresponding electrodes in this person's sensory cortex are stimulated.
Index, ring, pinky, index.
And just last year, it was shown that this actually improves the BCI user's ability to pick up objects, right? So they get faster and better at it. So the next step for the field in this domain is to restore not just a sense of touch, but also proprioception, which is our sense of where our body is in space. And proprioception is absolutely essential for making fast, fluid movements. Now, the challenge with this is that proprioceptive brain areas are buried deeper in the brain, and so it's much harder to reach them with electrodes, and we still don't have nearly as good an idea of how this sensation is actually encoded. But people will study this, and it will get done. And so the third new frontier, and one that I'm particularly excited about, is BCIs that restore the ability to speak. The point-and-click handwriting BCI I previously showed are remarkable, and they already go a long way to allowing people to freely communicate, even if they're unable to move or speak. But even that ultra-fast handwriting BCI is about 8 times slower than the speed at which I'm speaking with you today. So my lab here at UC Davis is working on a speech BCI that would decode the neural signals as a person tries to talk. Now, this is a much harder problem because we don't have an animal model for speech. There's no monkeys that talk. And so we know a lot less about the brain regions that are involved. But one way that neuroscientists have been able to get started on this journey is by taking advantage of human brain recordings that already happen for other medical purposes. So for example, people with intractable epilepsy often undergo a week or 2 of brain recordings in the hospital to help neurologists diagnose exactly where those seizures are originating. And these electrocorticography grids are placed on the surface of the brain and allow scientists to measure neural activity as the patients talk. So a few years ago, a group at UCSF showed that they could use machine learning to reconstruct what the person said from their ECOG measurements. So I'm showing you a spectrogram of the person's actual voice as they spoke out loud those 2 sentences. And here's what the researchers predicted was spoken from the brain activity. Now, it's far from perfect, it's not quite intelligible, but it does capture a lot of the broad speech features. So that was in a person who could still speak but just had epilepsy. Last year, that same group showed that in a person who has been unable to speak for 16 years due to a stroke, these speech brain areas also contained information about what the person was trying to say. So in this video, the researchers are asking the participant a question at the top, and they're decoding what words he's trying to say in reply. Now, this BCI is quite slow, and it was limited to only outputting one of 50 words. But this is still really, really exciting because it meant that the speech information is still accessible from the brain. We now just need to read it out faster and more accurately. And so my group is building on this work to develop a speech BCI that records from inside the brain rather than from just outside the surface. We're doing this because we expect that this will give us a much clearer picture about what the person's trying to say. So by analogy, if someone were standing outside this lecture hall, they could probably sort of hear what's going on. Maybe they can hear if I'm speaking or if people are clapping. But if you put a microphone right inside the room, you'll obviously be able to understand and hear the conversation much better. And so I got started on this a few years ago during my postdoc when I was still building BCIs to move robot arms. So as part of that, we already had these clinical trial participants who had Utah arrays, but they were in the hand area of the motor cortex. This is an area that's not believed to contain information about speech. But I thought, hey, these textbook maps of the brain were drawn using much cruder measurements than what we have available today. And so let's just give it a try. And our participants, who are wonderful, they're paralyzed, but they can still speak. And so they were very happy to just speak while I recorded their brain activity from this little patch of cortex. And so here's what I found. I'm showing you the activity of one neuron. Again, each tick is an action potential or a firing of that neuron, while the person spoke different syllables, which are shown in the different colors. And if we average this data, we see that this neuron is not only active, but it's distinctly so for different syllables. And other neurons, like this one, showed a different pattern of speech-related activity. So this is really cool, not only because it showed just how little we actually know about the human brain, but also because it meant that I could prototype ways to decode this activity towards building a speech BCI. And so here's an example of what we could do. I'm going to play an audio recording, and first you'll hear what the person actually said, and then you'll hear the same word synthesized from just his neural activity.
Veil. Garage. Tam. Shove.
Now, these are some of the best examples, and this is not good enough to allow a person to talk again intelligibly. But remember, I did this using measurements from basically the wrong part of the brain. And so what our lab is now gearing up to do, hopefully later this year, is to recruit clinical trial participants who cannot speak, and to place Utah arrays in what we think is the optimal part of the brain, the speech motor cortex. And I'm doing this together with my lab co-director and UC Davis Health neurosurgeon, Dr. David Brandman, who is here today, so raise your hand. Hey, David, thank you. And we're part of a 4-institution collaboration, which allows us to actually do this not just in 1 or 2 participants, but hopefully in more, faster. And as far as I know, we are the only team that is trying this approach, so I'm really excited to see where this goes. And so just the last topic I want to touch upon is the neural interface hardware, those devices that go inside the brain, and what's available to help BCIs, to develop BCIs to help people. So most of the work I showed you today uses this Utah array, and that is today the best available interface that's approved for human use. But it's actually pretty old technology. I mean, it's not quite 30 years, but getting there. Whereas, for just one example, today in research labs that study animal brains, many researchers are using a probe called the NeuroPixels. It was developed by a consortium of nonprofits and foundations only 5 years ago. And each of these little needles is not only smaller than one Utah array electrode, but it can also record from 384 distinct locations instead of just one. And these devices, which are made using modern semiconductor industry methods, are scaling up like you'd expect from silicon technology, right? Your iPhone doubles in speed every year. And similarly, in just the last 4 years, they've increased the number of neurons these probes can record to around 5,000 per probe. And so now several groups, including ours, are working towards adapting these devices for use in humans. And so while we're starting our speech BCI effort using the neural interface hardware that's already available, it could well be that in about 5 years, our BCI will look more like this, with more than 1,000 electrodes, fully wireless, and with a decoder running on a small device like a tablet. I am really looking forward to this, because at that point, I believe we'll have access to enough brain cells to restore really high-quality speech. And the form factor of the device is such that the patient could use it 24/7, at home, or out and about. And so that's the medium-term goal of our lab. We want to make it so that if someone is diagnosed with ALS or has a stroke and loses their ability to speak, we could actually tell them, yes, we have a therapy for you. We can place this device in your brain, we can pair it with a small computer, and now you can talk to your loved ones, you can call your physician, you can use a computer, you can write emails, almost as fast as you could before your injury. I think this would have a huge impact on the lives of these patients, their families, and the wider community, and it would also demonstrate to society how fundamental systems neuroscience, understanding how groups of neurons generate our behavior, like speech, can be translated into crucially needed therapies. And just lastly, zooming out, in the next 30 years, I predict that brain-computer interfaces are going to be used to treat not just paralysis, but also a wide range of neurological and psychiatric conditions, including memory loss, for example, from Alzheimer's disease, and currently untreatable depression. So this will be yet another tool in our arsenal in addition to those incredibly exciting methods that Drs. Kim and McAllister just talked about. So I think BCIs can make a difference in these other domains by precisely reading from and then fine-tuning the relevant neural circuits. And while I didn't have a chance to tell you about this today, there are many groups that are already working on this. All of those efforts will benefit from the much more advanced algorithms and neural interface hardware that are being developed and initially tested in studies like ours that are aimed at restoring speech and movement. And so it's a very exciting time to be in neuroscience and neuroengineering. So this work is only possible thanks to a large team of really smart and hardworking people who I want to acknowledge. They include our new lab, shown here as Zoom boxes, and my collaborators at Stanford and the other BrainGate sites. And then finally, a big thanks to my current and recent funding sources, and most importantly, these clinical trial participants. And these are truly incredible people who, despite everything they're dealing with, selflessly give their time to help us make discoveries and develop new technologies that will help in the future people that are in a similar situation to their own. So lastly, thank you for listening, and I'd be happy to take your questions.
That was really awesome, thank you. I just had a question on implanting these devices. Is there any negative effects of putting these things inside of someone's brain?
Great question. We should probably direct it to the neurosurgeon, but I'll give you the short version answer, which is, can you hear me? Oh, sorry, the question was, is there any risk or potential downside of implanting a device in someone's brain? And so, yeah, I want to emphasize it's not zero risk. The primary end goal of our clinical trial, because it's a very early stage, basically the equivalent of phase 1, is to establish safety. Now, to date, we've had a very good safety record, and that will soon be published with, I think now, 15 participants over 15 years. But it is a brain surgery, so there is always some risk. And so, really it's about balancing risks and trade-offs. If you're perfectly healthy and just want to control your smartphone with your brain, I would not recommend an implanted BCI. But also at the same time, there are a lot of groups working on non-invasive systems where they may not have that same accuracy. I don't think we'd be able to restore movement or speech with a non-invasive device, but we can probably use it for other diagnostics or sort of low bandwidth communication. Yeah, it's a great question.
Great presentation, Dr. Stavisky. I'm really curious where you place the module. I would assume at different parts of the brain you're using for the motor function versus the verbal function or writing function. How do you decide where that goes?
Yes, the question is, how do we know where to put the electrodes? And so, I kind of said we're surprised by how much we don't know about the brain, but we do know a lot about human neuroanatomy. So from tools like fMRI, from microstimulation during surgeries, from lesion studies, we have a broad map of which part of the human brain sort of is most involved in controlling different movements. And so if for our clinical trial to restore upper arm movements, we know that we go to the dorsal motor cortex, which is sort of up here. If we want to target the speech areas of motor cortex, we would go from the ventral motor cortex, that's here. There are groups that are trying to restore, for example, vision with electrical microstimulation, and we know that the visual cortex is sort of in the back of your head. Or if we're trying to write in a sense of touch or proprioception, we know that that's just behind that motor cortex or on the other side of the central sulcus. And so we kind of have the broad anatomy. And the other fortuitous thing is that it's not like there's little patches, it's not like, like there's one tiny spot where all of the information about the arm is, or all of the information about the tongue is. And if you don't land your electrode there, you're totally out of luck. It turns out that these signals are really widely distributed, and sort of, you can think of it as like, there's a hot spot where you have the most activity, but it rolls off gently. And so the more electrodes you put in, the more information you have about each kind of movement. And that really works to our favor.
Last question. All right, thank you so much, Dr. Stavisky.
Thank you.
Okay, so thank you so much, Dr. Stavisky, and to all of our speakers today, including our Data Blitz, for such informative and inspiring presentations. My heart is really filled with hope and excitement for the future of neuroscience and for the health and well-being of our community. As we continue to gain a deeper understanding of how the brain works, and especially as we continue to work across disciplines, you've heard of a lot of examples of collaboration today that make our work possible. We really believe that we're going to be making continuing and increasing transformative discoveries that will allow us to identify new treatments and potential cures for these disorders across all of these different diseases that you heard about today. At the Center for Neuroscience and at UC Davis, more broadly, we are deeply committed to this work and to improving lives. And we're really grateful for all of your continued interest and your support, and we look forward to partnering with all of you to achieve this shared goal. So now I'd like to invite my co-chairs, Dr. John Gray and Kostas Zarbalis, to come to the stage to join me in announcing the winner of this year's NeuroBlitz contest. And somebody is supposed to bring the envelope. Awesome. Okay.
Okay, we have a winner. And it is, for the NeuroBlitz presentation, Lucas Cuya, by a very narrow margin. And I don't see him. See. Oh, okay. By the narrowest margin imaginable, just one vote, but yet, just by one vote.
Oh, wow. Thank you.
Here you go. And congratulations one more time.
Terrific. So on behalf of the UC Davis Center for Neuroscience faculty, students, and staff, I want to thank all of you again for your participation, for joining us today. It's been great to be back in person. I can feel the energy. It's wonderful. We want you to stay healthy, and we look forward to hopefully seeing you again next year at next year's NeuroFest.